session one on next gen techniques for crop improvement. There will be around 11 presentations. Each will be 10 minutes, right? It's my honor and the pleasure to welcome Dr. Sendil Kumar Muttappa, scientist from NIPGR, that is National Institute of Plant Genetic Resources, New Delhi. He is not new to TNAU. He has already visited many times. Even 4 months back, he was giving a guest lecture to the Center of Advanced Faculty Training at Center for Plant Breeding and Genetics. And to tell few things about him, he is a distinguished scientist and researcher. He is affiliated to NIPGR and he did his PDM from Noble Research Institute, Oklahoma, USA. And he was the recipient of Woodward and Research Fellowship by the International Union of Biochemistry and Molecular Biology. And he was also the recipient of Ramalingaswamy Re-Entry Fellowship from DST, New Delhi. And he was awarded the DBT Innovative Young Biotechnologist Award in 2015, which honors the young scientists who have made remarkable achievements in the field of biotechnology. And he is the fellow of Indian Society of Plant Physiology and fellow of NAS from 2022. Sorry. He won the JC Force Gold Medal Award by the given by the Indian Society of Plant Physiology in the year 2020. He works primarily on plant molecular biology, plant stress and pathogen interaction and their genetics. He has made significant contribution to understand the genetic mechanism which underlie the plant growth and development, especially he worked on pathogen and plant interactions. And he is currently also involved in genomics, transcriptomics, bioinformatics, and research outcomes can be widely seen in publications. If you go to his website, you can see many publications which are published in very high-rated journals. And he also developed a database, two databases, SCLG database, which is stress combination for interaction in plants, and another is VGS database, that is virus including gene silencing database. These two databases are already developed by him. And again, if you visit his website, you can see many researchers were trained by him. Most of them are doing their postdoc in international. Everyone has settled well. Either they are working as assistant professor or scientists in ICR Institute, or they flew away to do postdoc in International Institute. So he is such a personality. We welcome you, sir, on behalf of TNAU to this AGSC. I also welcome Dr. Fogila, who will be a member for this session, PNH Biotechnology, and Jay Gandhan and Divagar, who is the student representative. I also welcome the oral presenter, but the students who come for oral presentation are also the participants. There will be around 115 posters, but I don't see anyone here. Only 25 students are sitting here. Tomorrow I will see. I will take attendance. I will see who are coming for the poster. So those posters will not be evaluated tomorrow. Those who are not sitting here today, we will not evaluate the posters by tomorrow. Is it okay? No. So again, on behalf of staff and students of TNA, I welcome Dr. Sendil Kumar Muttappa. He will be presenting a lecture, which will be followed by the oral presentation by the students. Thank you.
Thank you so much, sir. Now, I request the Director, Center for Plant Breeding and Genetics, to welcome the Chief Guest with the bouquet. I request Dr. E. Fogila Devi ma'am to welcome the Director, Center for Plant Breeding and Genetics with the bouquet. It's a great honor and privilege to welcome the esteemed speaker to the scientific community, a scientist whose contributions have significantly advanced the field of plant sciences. Here is Dr. Sendil Kumar Muttappa, the scientist from National Institute of Plant Genome Research, New Delhi. The scientist is dedicated to mentoring young researchers and helping to shape the next generation of the scientists in the field of plant sciences. Now, we welcome you, sir, to hear your plenary talk on harnessing innovation for climate-smart crop development. We welcome you, sir.
So, without spending much more time, and my talk, I'm going to keep it within 30 minutes, so that it will be very crisp. And there are enough resources, as Professor Ravikeshwar mentioned, available online, both my lab website and databases, that you can always go and visit for more information. So I will keep my talk as short as possible. So I just prepared my slides to address the actual content of this conference, especially the session that we are currently undertaking. So I will cover the techniques and tools that are currently available. First, I will introduce my institute. So we are coming under DBT, Department of Biotechnology, Ministry of Science and Technology, and our organization is part of BRIC. So in Biotechnology Research and Innovation Council, and this has several organizations like us, autonomous organizations, and those organizations, all of them offer a PhD. So that's why I specifically mentioned here that this is an excellent opportunity to do PhD in these frontier areas of basic and applied plant biology in these organizations. So please look for it. And we have PhD announcements coming frequently. Just now, one cycle got over. So not only NIPJ, but also all BRIC organizations, you can look forward for it. Normally, we know ICR, CSIR, and other universities of central and state nature, but these organizations are also another opportunity, very good opportunity for doing PhD, and also the training programs. Even at master's level, we do encourage based on the merit basis that you can also do thesis work there as a part of both MSc and also PhD. With that, I also introduce my research group. We work mainly on chickpea, that is contact correlate, and also on Arabidopsis to a bigger extent. As Professor mentioned, that our lab focuses on data science and computational biology tools like neural networks, ANN and CNN. We handle a lot of omics, including transcriptomics, proteomics, and metabolomics. And phenomics is also in a big way that we do it in the lab. We address two important questions. One, as he mentioned, the combined stresses, where the plant that when it is exposed to more than one stress, how it responds. And we also work on a concept called non-host resistance, where disease is only an exception, and most of the other plants are resistant to that particular pathogen, while only one or two are susceptible. So we are trying to understand why the other plants are resistant, that we call it as non-host resistance. And we use both physiological and molecular mechanisms to address to the questions. And we work both at the laboratory level on benches, greenhouse, and we have a lot of field-related studies, including at TNAU. So this is the privilege that I'm happy to hear. So this QR code will allow you to have my lab access and also the database access if you would like to go through it online. And first, I would like to appreciate, before I begin my talk, the entire committee who allowed me to come here and present before you, the PG Dean's office, PG Dean, and also the members who are here, who are involved in organizing this conference, especially the student friends. And with that, I will straight get into the talks. My topic is basically on climate change. This was also spoken well in the morning, the introductory sessions, both by Vice Chancellor and also other members about the climate change. So this doesn't need much of an explanation. We all know that this is an actual emerging aspect. And 13, that is the SDG 13 is the climate action plan under which that we also have recently published one article in Trends in Plant Science on this aspect. From there, I have taken these pictures. So this article actually enumerates how to combat, how to play a role in SDG, that is the Sustainable Goal Development. How to address this, how to contribute as a scientist into it. That's what we are addressing in that article. You can go back to my website and find out. So today, under this heading, I'm going to talk about the stress that occurs in multiple terms to the plants. That is more than one stress that comes and infects the plants and affects the plants, how to deal with that. And especially those technologies that we use, the new and novel technologies that we use to combat it. I'm going to take an example of chickpea all through my presentation. And I'm going to take an example of pathogen and drought coming together as a combined stress. So this pathogen is actually a root rot pathogen, and that causes a disease called dry root rot. And this is the combined stress. When drought and dry root rot comes together, it causes much more damage. But however, it is true for any of the stresses. When a host is there facing two, environmental stress 1 and environmental stress 2, this forms actually a triangle. Their interaction matters a lot. And this could be a positive interaction, meaning that it could be beneficial to the plant. One example for that is that when there is an abiotic stress, it's known that certain insect attacks are certain insects, not all of them, are going to be lower. Certain pathogens are going to be lower. So in that way, the plant could be benefited because of that. But most of the interactions are negative, meaning the stress becomes additive, like 1 plus 1, 2 stress becomes additive, and it actually, so 90% of the stress combinations are negative interactions. So the perspective that is needed in this is that we do always analyze single stresses in one go. For example, in all our studies, we focus on one particular stress, give that particular stress to the plant, and analyze its physiological, molecular, or other agronomic effects. It could be a pathogen from a biotic side, or it could be an abiotic stress from drought side. However, today's intention of this talk is to show you that these two stresses normally comes together. To a varying degree, maybe drought is prominent, but pathogen may be less, but they both are there together. The plant is actually responding to both of them. So the combined stresses are very common in nature. It can come under either abiotic category, abiotic biotic category, or biotic category. So these stress combinations are very important. And with that, I would like to just say that one of the important aspects to start this any work on this combined stresses is to identify the region. And that, for example, in Coimbatore region, what is the major combined stress that exists in nature? You can always analyze through a meteorological data, geoinformatics data, and also the common occurrence of reporting of pathogens and pests. So if you gather the data for about 20, 30 years and analyze it, you will be able to identify the region-specific combined stress. Once you identify, for example, in this map, using one of the data science approaches that we identified that Himachal Pradesh had reported 3.2% of disease incidents only, while Rajasthan, which had a higher occurrence of drought, had 41%. Concurrently, the Chhattisgarh, which had a very less drought incidence, for the same pathogen, it has showed only 9.1%. So the Rajasthan is the clear indicator that when there is a presence of an additional stress, namely drought, this disease incidence has gone way high up. So one has to identify a particular stress combination that occurs in that particular crop. So once you identify it, we have several approaches to target that, how to address that. So I'm showing up those approaches that we use in the lab. One key aspect is AI. AI is one of the common aspects that we are all now bound to use. So everybody must be prepared to learn some of the aspects that comes under this AI. One is the data science that we do, as Professor mentioned, that the SIP-DB is one of the contribution from our side from the data science perspective. And another actually is the machine learning based ones. We work on artificial neural network and convolutional neural network. I think both of them are familiar terms. One must be able to understand. You can always read the literature to identify what it is. And from the experimental approaches, we use these omics approaches. One is phenomics, and which is consisting of both image-based phenomics. We use RGB-based hyperspectral and thermal imaging, and finally we employ CNN to analyze the data. The second part of the omics is I'm keeping an example of disease assays. So we conduct experiments in multiple locations for the same trait. For example, lab, greenhouse, and the facility, that the sick plot that we establish actually in the conditions of our campus and the regular field conditions. In all of them, if you take an example of RGB, take the images from all of these conditions so that the research that we do in the lab finally can be correlatable to the field conditions. And such a huge number of images that, for example, if I'm taking an example of screening about 2000 genotypes. So I screen the genotype in lab, greenhouse, and the facility, basically a field mimic facility, which is a field facility, and then the actual field conditions. In all of them, we gather the photographs from all these aspects and then use CNN to analyze it. So when you have an annotation that you are coming out of such a large scale analysis, you will be able to actually correlate the actual inventions to the practical utility. And the second part is I will focus on genomics, then transcriptomics, metabolomics, and integratomics. Integratomics is another upcoming approach, which is nothing but integration of all these omics approaches to find out one trait, and that one trait is what contributes to the gene that contributes to the major gene that contributes to it, and it's the phenotype. So when we associate phenomics with genomics, transcriptomics, and metabolomics, that we call it as integratomics, this is again done through artificial intelligence. There are several softwares that are coming up in this aspect. You can read the articles on that. So in general, for the experimental approaches, we predominantly use an approach called gene silencing. As Professor again mentioned, that using virus-induced gene silencing is one of the key experimental system in our lab. And of course, gene overexpression system, which many of you are familiar with. And another one is an important one that is upcoming and also already in a big way started is gene editing. So this gene editing is also one of the new and novel tools that one should be aware of, one should be prepared with to use it for your studies. So first I will take data science approach, that how we develop the SIPDB. So this is the database. You can always get, you can just Google SCIPDB, you will get this database. It's freely available. Everybody can access in your mobile itself or in the laptop. The rationale is actually the data information is large. However, the knowledge that is gained out of it is confusing, or in other words, there is no linear interpretation of this data possible because the data is huge. So our strategy is to make it linearized, and that is done by using different AI tools. And those tools were employed under these three categories, namely phenomics, transcriptomics, and metabolomics. And these three offers a huge one linear scale information to you to get the actual interpretation of that particular huge data. So you can visit the database to know that information. This covers a large number of literatures, all peer-reviewed articles of close to 1,000. And we cover 123 stress combinations in this database, and we cover 408 organisms. Transcriptome of about 36 is already available in a bigger scale. And the genes that we identified so far from this database are around 45,000. The traits that we characterize is around 107. And this approach, the database was established initially by data mining and then bio-curated using the AI tools. And several programming languages were used to illustrate the interpretations, that linear interpretation that we get out of that. And then those repository, those were all deposited in the repository. Now this can be easily, the front end, what you will see when you visit the database is the retrieval and its integration. So you will be able to experience a seamless information flow of what you want. If you ask a particular question in that mind and browse the database, you will be able to get it. And that database is offering you out of so much of literature, which humanly impossible to read such large number of data and then get that information. So I will not get in much into this. This is actually the SIP database outline. You can again go to one of these QR codes. I would strongly encourage to visit our YouTube channel, which is very good in explaining all the database components. And we also present in Twitter and other components that we are routinely updating. So this will actually easily update you on what we do in this database and how it works. So briefly, this database involves literature mining and then public data collection, which the articles collected were grouped into several categories. And those categories were further analyzed using AI tools. And this AI tool is now representing its illustrations in this manner. For example, if you use this tree, if you go into the database, you will be able to see in a full scale, or you can analyze the trend, and you can also analyze the global distribution. So you can just click this global map and it will identify and give you the information according to the global location of what combined stress, where it is occurring, and which crops are affected, and what are the traits that are affected by this. If you are, for example, working in rice, you will be able to know what are the rice locations that the rice is cultivated, and what are the combined stresses that this rice is exposed to. And out of this combined stress, what are the actual traits in the rice that are actually affected in terms of starting from yield to all the attributing traits. You will be able to know that. I will quickly go through the next one. We use machine learning, as I mentioned, both the artificial neural network and combined convolution neural network. The first thing I would like to show example is the artificial neural network. So the database that I just showed you offers the information in these three major categories, and a particular traits importance can be obtained by selecting those data sets. You will be able to cull out and then select the data sets that we subjected them to artificial neural network machine learning approach. Then we are able to categorize biomass, yield, and growth, and then build the model. And now this model can occur. I'm just giving one illustration of it. How each of these combined stresses is connected to each of the traits that are contributing to the yield and yield attributing traits. So this is a very complex one. However, it is interactive with the database. If you actually go to the database, you will be able to see each one of them. You can pull out and then see what is the actual correlation between these two. And these all data sets, what is offered in silico, were practically validated in our field and the other situations that I explained, laboratory and conditions. And this model has clearly showed you a tight correlation between the predicted and the actual values. Meaning, this indicates that the database information that is in silico predicted is highly correlatable to the practical relevance. For example, in this particular case, the particular conclusion matrix that I am showing you is actually from rice, and for a particular stress combination, we are trying to come out with the major stress combination for rice as drought and heat together. So that's the correlation that we are able to see. So such conclusion matrix, you will be able to see a large number in you will be able to generate one in that SIP database, and it will be able to help you to understand the actual traits that are impacted. So the next one is convolution neural network, and I'm not going to get into the details of it. Most of you who are from bioinformatics background will be knowing about it. So these are mainly used in my lab for image-based analysis. As I was telling you, in all those 4 locations, we have thousands and thousands of images for that particular disease and combined stress, and we use this flowchart to actually run the images to identify the patterns, and hence will be able to predict and assess the genotype performance under the particular combined stress. So this input image is converted to a convolution layer, and then pulling up this layer, and then fully convoluted layer is actually the one that is actually offering the CNN output. So this model, what we are developing and also developed previous ones are available in GitHub. So I will not spend much time in that. You will be always going into my lab accounts GitHub files. You can download it. It's free to use. And once the model is there in your hand, you can actually download the datasets from our SIP database. And those base datasets, you can employ the model and then use it further for your own research purpose. So the second one, the next one I would like to focus under the phenomics is the imaging part. So I just want to spend some time to explain what is the difference between RGB, hyperspectral, and thermal imaging patterns. So you all know RGB is based on the regular photographs that we have. Hyperspectral is a little more advanced, that it actually captures more finer details than the RGB. Thermal is based on the temperature related measurements. The advantage here is that the thermal ones can allow, for example, if you are thinking about a disease, before even the external symptoms are visible in the leaf or root, the thermal readings will be able to show you. Just like how in the COVID situations we use thermal cameras to detect the temperature or health of the plant, same thing you can do it with the plant before even we see it. And once the microscopic cell death or microscopic events are happening, at that time we can't see it in the naked eye. The hyperspectral cameras will be able to actually capture that. And then, of course, RGB, when the extent of disease is highly visible, which can be photographed. So these are the 3 type of image categories that we capturing, and again use CNN to analyze those images. Now you can imagine in our lab, these 3 categories of images are used for all the lab, greenhouse, mini field conditions, and field conditions. So there are 4 environmental situations. We gather the images under these 3 categories. So you can imagine the number of images that it will finally accumulate to, and then use the CNN based system to analyze it. So it can give you information that is starting from initiation of disease to the culminated disease, that what is the degree in which that it is progressing, and also across the multiple environments how it works. So it gives a very standard data set that allows you to have a very powerful model that you can develop to predict and assess this. So the advantage here is that, let's say if we have 5,000 chickpea germ classes which we are currently handling, you'll be able to screen and make a data out of those 5,000 lines very digitally, which is very useful for Jivas, because Jivas power depends on the phenomic data that you supply. So if the phenomics data is very good, then your Jivas results are going to be very good. So currently we are applying that to get the data. So this I have already explained. As I said, that RGB is the regular camera. Hyperspectral cameras can be used for throughput, strategic of growth and disease detection, and also the thermal ones that I said will be useful for early detection of the diseases. Of course, this is not only for disease. Since I'm taking an example of disease, I'm showing that, but it is also true for drought or any other stresses that it will capture the plant health overall. So this is one image that I would like to just project generally to the students. If you use a microscope, this is what the dry root rot will look like. And if you use a root scanner, this is how the root of 3-dimension will look like. And this is the regular camera images. And these are the mini field facility that we have on campus that projects the overview of the image actually. So if you have this plan of particular 9 plants per hour based on the RCPD system, you'll be able to get the photographs like this across the growth regions. By the way, these images are taken every alternate day. So you will accumulate that many number of images as the plant grows. And the other one is to use drones in the fields to capture the images. This is the drone image of the dry root rot incidence in one of the field near Kadapa, Uttar Pradesh. So this clearly indicates that the disease progression. If I play a movie out of this, which currently I don't have, we will be able to gradually show you how the disease has progressed and how the area has been extended. And the estimation is very easy in the field conditions. I'll just spend 1 minute in explaining this RGB image processing system. Basically, these images that are captured through these mini-sig plots, where you get this kind of images, are now trained, used for, basically used for initially the training of the machine. And then these data sets are initially augmented. After the image augmentation, we use the test image data sets, and we pass through this training process with large number of images. Finally, the masking is done to make sure that the non-disease areas are clearly learned by the CNN to make sure that this is identifying the right disease at the right proportion. And the outcome that is coming in the term of matrix, confusion matrix, shows you the extent to which how the model is powerful and how it predicts, it gives you the actual values. So I will run through it quickly, the hyperspectral imaging. So this again, actually, you probably know that from the visible to infrared, what is the spectrum? This hyperspectrum is able to cover the entire range. That is why it is better than the RGB images, and it is able to develop, it allows the CNN to develop multiple layers more than what it can do it for RGB images. And such layers are very powerful in getting the final outcome in a very precise manner. So if you be able to distinguish two closely related diseases which are symptomatically same, but this will able to distinguish the diseases very clearly out of compared to RGB images. This thermal imaging is much more powerful. These thermal cameras actually scans first, condenses, and then uses the detection strategy, and then amplifies and then displays. The same image that what I showed you for the RGB and hyperspectral is now looks like in the thermal camera size like this, and the control plants are like this, and you can see that the dry root rot infected plants which are showing like this are the infrared, of course, in the early stages. It shows the images like this. You can clearly see the distinction between the control, that is the CTT values are much higher here, whereas this infected plants shows the very less CTT values, which is a clear indication that the leaf water status is less, and the other factors that are influencing the plant is much drastically affected by the pathogen. The other one, I just want to show you the disease screening assays. This is true for any stress-related aspects. I'm just showing it for diseases under all the 4 conditions that I displayed to you. In general, under laboratory conditions, we use the pouch method. You can see that the same number of genotypes that we have screened shows the normal distribution of disease severity index. This is also reflected in the form of a disease severity score. And we can also clearly see that the first necrotic area identified by the image analysis is also matching with the necrotic area that we actually scored for. The second approach that we use in the lab is, and in the greenhouse is the, I just want to introduce this term rhizoboxes. These are very commonly used these days to detect the root system, visualize the root system when it is actually in a live plant. So these boxes, as you can see here, are the ones that are actually the, they have covered it because they want to avoid the light to the root. But you can open up this black area, and this will look like something like this, where you can clearly visualize as the root grows. You can picturize it using the model. If you subject it, it will remove all the soil particles from its events and take only the roots. You can see only the roots in the computer. And such root systems can be easily observed for many of the parameters, including the disease ones that I described it. So you can clearly see that the disease score that are scored by this model out of these box images are really good. And you can see all those parameters that you cannot see the naked eye in these pictures, which are very useful for image-based analysis. So these rhizoboxes are picking up very well. It is very easy to customize and then use it for your own experiments whoever is involved in group-based studies. The other one is building facilities for mini field conditions, where before you go to the field, you will have to make sure that the data that what you are getting is analyzed in the mini field conditions. So this is one of the facility that we have developed to assess the dry root rot and drought together combined stress in our campus. So this actually helps us to gather the images in multiple views, basically the aerial imaging and also the individual root scanner based imaging. I think if the video shows, we are also having the cameras in the facility itself that actually using these images will be able to picturize the plant growth and also how the changes between genotypes occurs in this with and without disease. And these video images are again used for semen-based system to know more about this plant in terms of its growth habits and disease related features. In the field conditions, we have a regular field plots, and these images that we are gathering can be obtained from the low-flying drones or high-flying drones, depending on the need of that. And those again, the drone images are again used for further analysis in terms of identifying those performance of those genotypes that were undertook under the mini field and greenhouse and laboratory conditions. There are other 4 approaches that I will quickly go through before my time ends. Is based on genomics, transcriptomics, metabolomics, and the integratomics. The first one I would like to know, and I think this is known to many of you, is the genomics. The basis for this GWAS is, as I again emphasize, that it is the phenotypic data, that how robust it is. So using those previous methods that I showed, we were able to get a very good tight phenomics data, and our genome-wide association mapping is really coming up very well. And we have identified genotypes that are resistant and susceptible in this regard. So I just want to introduce the power of this genomics techniques whenever there is a good phenomics data that is available. Again, transcriptomics is another important global approach tool. So in this, we are able to actually identify this interactive heat map, which you go to the SIP database, you will be able to find it. Clearly shows how many of the shared responses that the genotype had, and also how many of the unique responses under combined stress that it produced. And also we were able to map the genes and its pathways that are related to it. This is, I think, most of you are aware of this genomics, the transcriptomics part. So I will. The advantage with the SIP database is that this transcriptomic part, which is inbuilt, has also a blast tool involved in it. So even if you don't have a species that has a good transcriptomic research, you will still be able to use that for your studies. Next one is metabolomics. I will again quickly go through it. So this is again a vast area which metabolomics can be done for many things. Either it could be for hormones, it could be for individual metabolites or non-targeted metabolite identifications. So you can do it for multiple aspects. I just want to introduce that this is one of the strong omic approach that one should be able to use it. When you correlate this metabolomics with proteomics, transcriptomics, and metabolomics, using the next approach that reveals the pathways, that is called integratomics, you will finally get a very strong candidate gene or a pathway that you will be able to further work on it. Again, if you go to the SIP database, you will be able to get this transcriptomic and metabolomic datasets. I'm just showing only for 2 omic approaches. If you follow a particular pipeline that is suggested here, again, you don't have to worry about this slide. You can go back to the database. The same information is available. This is just an example to show that how we identified a gene called asparagine synthase 1 for that particular for its role in combined stress responses, and where that gene particularly places in the overall pathway. That again, it can be illustrated in the database itself. So this is called integratomics. It is completely based on integrating multiple omics data using a particular conversion point. It's not the one particular software that does, but it's the pipeline that matters that you can use it. And this is again available in GitHub files, so you will be able to download that and then use it. Of course, this is something known. Once you know all the omics, which all use broader information of multiple behavioral pattern of genes, ultimately you have to really test its function. And only then you will be able to know whether that particular gene is directly relevant to a phenotype or not. So we use gene silencing based approaches, gene overexpression based approaches, and in very recent terms for the past few years, gene editing based ones, which is now very popular, and the precise editing is possible for the traits that we wanted to manipulate. I will quickly go through it. This integration of these techniques, whatever I described earlier, is the one that we were able to come up with to finally come out and identify a resistant cultivar that we were able to use it using multiple approaches. So we had a germplasm of about 5,000 plus genotypes, and the strong phenotypic identification, phenotyping of the previous approaches allowed us to have a very good phenotypic data that is also reflected in the form of the database. And we were able to use multiple other tools that I told, the omics approaches, including genomics and other approaches that I told you. And finally, we were able to use the genome editing tools and identification of genes from the GWAS analysis. We were able to have certain genotypes that are tolerant, and we were also able to come up with the plants that are moderately resistant to dry root rot disease. 2 or 3 other important concepts I just would like to know that I think the speed breeding was spoken about in this morning itself, which is a very important concept. This is called rapid generation advancement. You can always go back to my YouTube channel. We have a very good program on chickpea for reducing the duration from 120 days to half of it. There are illustrative presentations available. So this speed breeding is one of the important approach that it picks up. So the basic principle behind is that how you can use light, temperature, and few other environmental factors to alter it in such a way that the plant completes its basic vegetative phase very quickly and enters into the reproductive phase, so that the seed that is formed is much quickly comes before the actual duration is complete. The second important concept which all students should be aware now is about the rhizospheric engineering. It is now coming up in a big way. You just go to a science cell or any of those papers, every month at least one paper will come on this rhizospheric engineering, which is nothing but engineering the microbiota in the rhizosphere in a way that is compatible with the plant, which is basically a part of sustainable goals that SDGs that they have developed. Instead of applying chemical fertilizers and other means of chemical aspects, how we can actually gather the microbiota around the rhizosphere, both the genotype that is receptive to that particular microbiota and also the microbiota that is compatible among themselves in such a way that it is having less number of representation of pathogens, but more number of beneficial aspects present in that. So we were also able to show one example of this microbiota by identifying one of the AMF, that is orchestral mycorrhizal fungus, with one of the rhizobial part with another bacterial system called Rhodostomonas as a combo that perfectly fit into solving dry root rot under field conditions. So this is one aspect, rhizospheric engineering, you may want to remember. The other one that is picking up, which was there earlier also, is called bioirrigation. The concept of intercropping along with the application of AMF. Actually, this allows actually the fungus, the AMF fungus, to harvest water from one of the, let's say you have a system of pigeon tree with another a deep-rooted pigeon tree system with another one intercrop. The intercrop ones which has a shallow root system will get water through the network of it. It's called mycorrhizal network. That network provides water to a cellular specific. It's like targetedly irrigates the cell directly. The advantage here is that the water loss is less. Second is other pathogens like this macropomina and others which I spoke about that cause dry root rot are dramatically reduced because those pathogens tenses the turgor of the cell and then thereby initiates the infection. However, if this cell turgor is maintained by these mycorrhizal fungal systems by this network, from a deep-rooted plant harvest to basically lifting of water and providing it to the neighboring plant. So this is also picking up as a big way, and it is all being incorporated as a part of rhizospheric engineering. I think I will close with that. The summary here is that if you will be able to employ all these parts as in an integrated manner, you will be able to develop a climate smart, in this particular case, chickpea. And our lab has done it, and hence I put it's from my own experience, these approaches and its integration. With that, I will stop, and I would like to acknowledge my entire lab and also grants from multiple agencies for giving me time. If you would like to ask questions, I'm happy to take.
Thank you so much, sir. We are fortunate to listen your inspiring lecture and the research with us today, sir. Thank you so much for giving the clear that your company stress interactions for both biotic and abiotic, and the different omic strategies to analyze the disease levels, and the usage of machine learning approaches, databases for the data analysis. Your view on laboratory assays will be definitely useful for us, and we will try to implement in our research, sir. And now it's time for our student friends to have an interaction with the.
Yeah, we are running out of time, so it will be good if you quickly ask them. Yeah, go ahead.
Questions on 3 phase. So So there is another screening sheet called paper plot assay.
Yeah.
So in that assay, we are maintaining a temperature of 28 degrees Celsius. But in many paper, they are mentioning that this macrophomina is particularly destructive under high temperature.
Yes.
So will this 28 degrees Celsius be enough for the pathogen to cause?
Very good question, but under the laboratory conditions, unfortunately, we increase the temperature, although you will get very good disease symptoms, but the plant performance by other means is reducing. Whenever I call it combined stress, when each of these two stresses comes in an equal way and then affects the plant, it's fine. But if one stress comes in a way that already predisposes the plant, then introducing macrophomina is not going to be useless because you are infecting an already dead plant. So yes, if you are managing to grow, if you are able to grow chickpea under let's say 32 degrees centigrade and then infect macrophomina, that would be the perfect assay. However, since chickpea is not cooperative under 32, we are trying to bring down the temperature to 28 and then doing it. By the way, chickpea is a winter crop. Normally, in northern states, the chickpea is grown somewhere between 8 degrees centigrade to 10 degrees centigrade. That's the temperature it likes to grow. Although in our conditions also it grows, which is around 20 plus. But that is the condition in which the chickpea is optimal. So in that way, if I consider 8 and 10 to 28, it's high temperature only. So for chickpea, 28 is already a high temperature. However, I could not increase more than that simply because of this reason. Ideally, it could have been good, but it's not feasible.
Another question is
why
you are preferring sick plots over sick pots?
Simply because in many of the pathogens, not only this macrophomina, but many of the root pathogens doesn't have a uniform infection. You can look at the disease incidence in the sick pots, it will be always non-uniform. That is simply because of a lot of factors that this pathogen actually is becoming conducive is possible only under a large plot conditions where it experiences. So they could come from lot. We have one paper that is on ANN based assessment considering nutrient dynamics. So this fungus, although the destructive nature is more under those conditions, but for experimental conditions, you want that destruction to happen. So if you want that to happen, the big plots are the only ones that can provide the dynamics. So only when the dynamics exist, the pathogen is aggravated and infects more, and which is very difficult to simulate under the sick pot conditions, under pot conditions. However, it is not true for every disease. Certain diseases are as destructive as field conditions in the sick pots also. In those diseases, you can use sick pot. But with specific reference to chickpea diseases, namely dry root rot, weed, black root rot, collar rot, all of them sick plot. The question is fine, like in terms of definition of a particular method, technique.
So I have a doubt, sir. What is the size of the training population you have used for a database?
So this database is not just our own result. I will divide this answer into two parts. Part one is to straight answer your question. For chickpea, we have around 5,000 plus, and this 5,000 plus is now divided into 4 treatments: control and drought, pathogen, plus combined stress. So we have 4 data sets. And each one of them, depending on the method, we have replicates. For example, we followed 4-step process of evaluation. The laboratory ones, each of those genotypes and under each of conditions will have 8 to 10 replicates. Under the greenhouse conditions, it will be around 3 replicates. Under the sick plot conditions, it's based on the design, you might have seen the pictures, 9. And under the field conditions, it's the defined plot. Sometimes it ranges from 25 plants to 30 plants and stuff like that. So now for each of them, we gather, if it is an image or a direct data in numerical data, we gather for each of them. So that is the amount of data sets that we are getting in. So this data set is the representation for one particular genotype for that particular treatment. So now you can imagine the scale of that. This is for my lab work in chickpea. However, in the literature, it is difficult to expect all such data sets systematically gathered for one genotype. So in the literature, the database host could range from minimum, we managed to keep 3 replicates. However, most of the cases, it could range somewhere between 3 to 10, 12. So that's the replicate range that in which that it will cover. So the data set number is going to be less in those literature ones. But since they are all picked up from peer-reviewed literatures, it must be fine enough to interpret from that small data set.
Okay, so thank you, sir. If there is no more doubts, we thank you so much, sir, for the nice session, and it was really useful for us. And now we'll break for 10 minutes for refreshment. I request everyone to assemble as soon as possible after the refreshment. After the refreshment, the oral presentation is about to start. Refreshments are arranged as a regular convocation.
We will start our session.
A warm good afternoon to the oral presentation session for the Theme1, the Next Gen crop. And once again, we will explain the rules for the event. A participant for the oral presentation is given with a total of 10 minutes. At the 6th minute, we will be giving a warning bell. And at the 7th minute, we will be giving the double warning bell. After next 3 minutes will be the discussion. And the mark split up is the content of the presentation carries 10 marks, clarity and flow of presentation 10 marks, novelty and contribution to the field 10 marks, ability to interact and discuss 10 marks, timing 10 marks, and the total of 15 marks. Once the participant exceeds beyond the time limit, 0.5 marks will be reduced. And the first oral presenter, Mr. M. Durga Prasad, on the topic genetic analysis in rice for logic resistance.
Good afternoon, everyone. On the dais and off the dais. My topic is on genetic analysis for logic resistance in rice. So we are well aware about winds, storms, and the yesterday only Tungabhadra Dam, one of the gates got broken. So the question is, how do we quantify the lodging? I mean, how do we phenotype the lodging? So how to move this slide? So the question is, how do we phenotype the lodging? Because it's very much difficult to phenotype. So this is an instrument known as universal testing machine, which is used to phenotype the lodging resistance. So we have to take a cull, we have to keep between the two fulcrums at 4 centimeters distance, and then we have to fall a load on it. Based on how much a cull can resist the load, we can quantify in terms of bending stress. Apart from that, we are having something known as section modulus, which is nothing but it's a geometric property of the rice cull, which includes both outer diameter and inner diameter. So the multiple of this both only is bending moment and breaking with the unit of gram force per centimeter. And then we are having cellulose crystallinity. It is very important to measure cellulose crystallinity because it will tell flexibility of the rice cull. Whenever the cellulose microfibers are very much orderly arranged, it leads to high strength. But at the same time, the cull won't be flexible because of high crystallinity. So we want low crystallinity for a rice to be lodging resistance. Low crystallinity means so all of these parameters was measured in the of 30 germplasms, which includes cultivated varieties, land uses, and also wild relatives. And then to understand, there is one phenomenon known as bending and breaking. We have to understand it. When a plant is too tall, it is usually susceptible to bending type lodging. But when a plant is dwarf, it is resistant to bending, but it will become susceptible to breaking type. Which means when a dwarf plant will usually there is less expression of genes for lignin, cellulose. So in our study, I will just report like as the plant height is increasing, the column strength will increase and section modulus also will increase. So to understand, to phenotype 30 germplasm was taken, and to understand the gene expression for tall versus dwarf, wild relative grandiculomis and the cultivars were taken for transcriptomic studies, and PPT 5204 dwarf variety and 436 the tall variety was taken for transcriptomic studies. So if we see clearly, it's all known strength. There was a clear strength of the tall varieties were more. Section modulus of tall varieties was more compared to the cultivated and land varieties. And also, what is the degree of leaf sheet here we can say degree of leaf sheet reinforcement at this end. So it is nothing but we are measuring the strength, including the leaf sheet and without the leaf sheet. So suppose for any cultivar, leaf sheet is the contributing factor for lodging resistance. We can understand by DLSR. So whenever DLSR is more, it is nothing but leaf sheet is contributing more towards lodging resistance. So even in our study, what we have got is the cultivated varieties were for cultivated varieties, leaf sheath played a very important role for lodging resistance, for the high culm strength. But in land races and the wild relative, the two wild relatives, there was no much importance of leaf sheath for high culm strength. So, and this is the cellulose crystallinity, as I told at the starting, it tells flexibility of the rice culm. It was measured for cultivated varieties, also land races, and also for wild relatives. So, if we see whenever the cellulose crystallinity is high, it is not good for lodging resistance. So, our wild relatives were found to have cellulose crystallinity of 49 and 44. And the habitat, which is one of the renowned lodging resistant cultivar, was also found to be high with cellulose crystallinity. So we have to select a genotype which is low with cellulose crystallinity, like Gandhasala, landrace, and 436 cultivated variety were found to be low cellulose crystallinity, which is good for lodging. And we have used a novel MGIDA index for ranking the germplasm, the 30 germplasm. So we have set a idiotype. So in MGIDA, we will set a idiotype. And then based on the idiotypic scoring, the RG130, 436, and Karupakauni. So this is the order of the germplasm which is found in terms of lodging importance, lodging resistance importance. So anything which is within the circle, they are lodging, they are not selected. Anything outside the circle are selected. So this land is Karupakauni, Akshagan, and this Go56 and RG130, these are selected based on our set idiotype. This is a metroglyph studies. So it is clear that all our cultivated varieties mostly fall under less bending stress category, and their plant height, even though it is less, obviously there is a trend. So if we see these land races and these wild relatives, even though they are tall, their bending stress is more and their section modulus is more. So, so that is why we will say like even a tall plant can be made lodging resistant on certain conditions. That is, cellulose crystallinity must be less and bending strength of lower internode must be more. Then even a tall plant can be made lodging resistant, and Chinese have already developed many tall varieties which are lodging resistant. So as I told at the starting of the presentation, tall versus dwarf and cultivated versus wild. We have to compare the gene expression to understand why we are saying that tall plants are having more strength and dwarf plants are having less strength, current strength. So to understand that, we have used BPT5204, and this is the workflow in the previous slide. So we have used BPT5204 and 436 as a combination, and also grandilonis with cultivated varieties as a combination. If we see in this phenyl propionate pathway, it is very clear. Like our this peroxidase genes are very high in expression in our tall variety like 436. It was several folds high, and in dwarf varieties like 436, peroxidase genes actually are very high in expression in tall varieties like 436 when compared with BPT5204. I think there's a mistake here. Actually, I'll just re-interchange. BPT5204 was found to be less with the peroxidase genes. And when compared with the wild relative and cultivated variety, we have found that the high strength seen in Oryza grandilonis is not because of the cellulose, it is not because of lignin. Because if you see in this pathway, Oryza grandilonis is showing less gene expression for even CCR, also for CSC, and also for peroxidase also, it is showing less gene expression. But the next slide, if you see for cellulose synthase gene, Oryza grandilonis is very high in gene expression for most of the cellulose synthase genes. Like 4, 7, and 9. 4, 7, 9 are contributing for secondary cell wall, and the 3 and 2 more are contributing for primary cell wall.
Time of participant.
So this is the phenotyping figure to just validate the genotyping. So lignin, fluorochromosomal staining for lignin staining. So in Oryza grandilonis it is less, but in our 436 it's more, and BPT52 is more. Thank you.
Please be prepared. In the meanwhile, I will ask a couple of questions, but somebody should ask from there also. So the first question I have is, we know that lignin contributes to strength, right? And in the pathway that you have shown, the count, if you silence count, do you think the plant will be lodging resistant?
So can you repeat?
COMT, if you silence that gene, would that plant be resistant to lodging?
Sir, we have not told anything about COMP. COMP was not.
We shown that there is a correlation between lignin and its strength. Right? Is that true?
No, sir. We found like this is very much complex question, a complex answer, because we cannot say lignin alone is contributing for.
Let's say lignin is one of the factors that can contribute to the strength. right? So in that case, if you carefully, since you have done the pathway, whole pathway, if you silence one of the COMTs, the resultant lignin, whatever that it comes, whether it is S plus D or whatever the ratio in which that it comes, would that result in a theoretical sense, launching resistant plant or susceptible plant?
Cannot be told.
Okay.
Because in our study, the grand clue is lignin gene expression is less only, but still strength is more. So we were thinking like it might be because of high cellulose and high cellulose crystallinity. Because lignin gene expression was less for in that pathway for most of the genes, lignin was less compared to 436.
Okay. So I'll just tell the answer like although lignin is a complex polymer, S by G ratio decides the strength. Okay. So silencing and overexpression will have a difference in lodging versus non-lodging. So my second question, unless somebody else has, is this Karupu Kavani is popping up in every possible studies, and it popped up in US studies also, seemingly performing well. So what is that not just related to your trait that this Karupu Kavani has, a list of those parameters that this is able to come up with many of the past two traits, that it becomes an example in any of those presentations.
Yes, Karupu Kavani, the landrace is very much good even for its therapeutic properties in its grain, like flavonoid content.
Why, what is that is there in it that it does? There must be some common features that it is able to exhibit, and hence it's popping up in whether a person who is working with lodging can also show that, another person who is working with disease can show that, a nutrition perspective can show that. So what are the things that you would expect that landrace has?
Nutrition properties are good. It is resistant to.
I know it has good qualities, but I'm asking why. Or what contributes to it?
Usually our the primitive land races, all of them are hubs for most of our hotspots and all. So now people are concentrating just to utilize them for introducing those genes into our modern cultivars through marker-resistant backgrounds. Even like most of our land, like Katalat of Assam or Sauli or
I know they do it, but what is that the plant is able to have? What does it have? Okay, thanks.
Since from the student side. So it goes here. Thank you. Thank you.
thank you.
Thank you, participant. Next, I call upon Dr. V.J. Shobana on the title Automating Chromosome Scrolling Using AutoML Models for Identifying Functional SNPs and Indels. Case studies in cucumber and soy.
Distinguished panel members and my dear researchers. Here I am before you to present my topic on distinguished panel members and my fellow researchers. Good afternoon, and my topic here is automating chromosome scrolling using AutoML models for identifying functional SNPs and indels. And I'm going to also share the most successful stories in cucumber and soy. First of all, this will be a new terminology for you all. Chromosome scrolling. It is nothing but you scroll across the chromosome. This technique was developed by our team, and this is the manual screening of a whole genome of a plant. It could be any other organism also, and it is a chromosome-wise scrolling in a particular database called Pathway Genome Database, which is developed with the help of the pathologic algorithm. And here you need a GBS data set and the identified SNPs and indels, and you're going to take those SNPs and indels and search whether those SNPs are present inside the genic or non-genic regions of the genome, the organism's genome. So as I said, here we have the SNPs generated from the GGS dataset, and you're looking for a direct hit or an indirect hit. Direct hit is an SNP that is found inside a genic area. An indirect hit is one that is found 500 to 1,000 base pairs before or after a gene that we are interested in. And so, since this is the Pathway Genome Database, it is already having a lot of information from various other fully annotated organisms. So even if you don't know the function of a gene or a genic area,
genomic region, you can refer it with that of other organisms that have been annotated already, and then you can quantify it using RT-PCR. So here we had two methods. We either go from the chromosome to the gene or through the pathway and backwards to the gene and find out the position on its chromosome. And so we can find out the pathways that are related to the genes of our interest. Here we have, I have a little video on it. I'll play that last one because for the want of time. And here the first case study is, we studied the gynaece habit in cucumber. Here we have published this result using chromosome scrolling in 2023. We found out 74 direct hits, and with the help of the cucumber genome database, which is also having the cuckoo site, which is the pathway database of cucumber. And here we had two parents and their F1, and we found out the ethylene synthesis gene ACS1 and SNP found directly inside that particular gene. So it is found in chromosome 6, and also an allele that is related to histone N-methyltransferase, which is related to the gynaece habit or female flower production. So these are the direct hits that we have identified, and these two genes have been again found to be directly correlated with gynaece in cucumber. This is the paper that we have published. And here we found out two pathways, and if you go in the pathway, pathway database of cucumber, we have this ethylene biosynthesis pathway and spermidine biosynthesis pathway. Here we have two compounds that are related to this gynaece habit, and we have all the related genes that are correlated to this pathway. So if we go for any specific gene that is very directly proportionate to the expression of this character, we can find a marker and this gene as a marker for us then. The second study is the extended shelf life in cucumber. That is, it took me 2 to 3 weeks for doing scrolling for gynaece in cucumber. And when it comes to extended shelf life in cucumber, it almost took me 2.5 months. So even in a plant like cucumber, which has only 7 chromosomes, it was very hard because this extended shelf life character, it is related to almost 44 genes and many other associated phenotypes that are associated with this particular extended shelf life in cucumber. For our surprise, the same gene 1 amino ACS1 has the same contribution for extended shelf life also as in the case of gynaece. So having spent so much time on chromosome storing for cucumber with 7 chromosomes, when we thought of using this technology in sorghum for finding out the biosynthesis of iron accumulation, we found out we went backwards, that is from pathways to chromosomes. So here I found out a very important pathway called heme biosynthesis, and this comes inside the metabolism of porphyrin metabolism. Here, since I was looking for any Fe2 plus or Fe3 plus ion that is directly correlated with the accumulation in the grains, I found out two pathways, ferritin degradation and penton reaction. So this could be a major hint of how iron is accumulated in sorghum grains. As you can see, heme biosynthesis has an end product of Fe2 plus. If we target any one of these very particular significant genes, we can find out how the accumulation of iron happens in sorghum grains. This is actually found out by means of a process called ferrooptosis, that is programmed cell death. And this process has been widely used in cancer studies. That is, the iron, Fe3 plus iron that is released here is used to rupture the cell, that is the cancerous cells. So we can go deep into the study and find out the basic mechanism behind iron accumulation and also zinc accumulation. So automating the chromosome scrolling by use of AutoML tools, machine learning is selecting the right machine learning algorithm to tune into the hyperparameters. Since it is a daunting character task for humans, we can skip those model steps in machine learning and go for AutoML. So for AutoML, many studies have been performed, and T-POT is the one that we have studied here. You can see this is very daunting when it comes to machine learning. With the phenomenon of AutoML, you can save time, use it easily, it is scalable, reduced bias, and also you can train a data set as we have said, and we find out the best model for hyperparameter parameter optimization.
If somebody get ready to ask a question, in the meanwhile, I'll ask her some questions. So you just introduced the last part, that should have been the most elaborate
Yes.
part. Should have covered that, that's one comment. But by the way, so the cucumber genome, is it of the reference genome quality?
Yes, sir. It is, they have version 3 of the Chinese variety DOM, which is the recent one that has been sequenced in
So
2009.
that's good assumption. Okay. And the computational tool that you was at the end in the couple of slides that you were trying to explain, what are the competitive other tools that are available that can do the same
Yes,
one?
sir.
Can you name some of them?
It is H2O.E type, and those are based on scikit-learn.
So how would you compare that with your?
That is, they are used for, I have referred a paper where they have standardized T-POT for omic studies, integrated omic studies. So here we can have, it can automate the major daunting task of machine learning. The iteration is very easy, and even without any programming language, the machine can do that iteration and find out the most best part of you have various models. So with the training data set, it can do the selection of the best.
I understand the application, but what is the basic script to start this teapot?
It's a Python.
Okay. And you explained that E3 coming up in your predictions is expected and it's well correlated with the shelf life. It's understood. I would really want to know what is the step that you identified, because I'm not seeing that, because you are citing safe.
Yes, sir.
You could. So what is that step? So those normal expected pathways like ethylene in shelf life, it's a good indicator that the model is doing well. But what is the outcome? Like where
So
is
that is what we have this method standardized chromosome slowly, and we have published it. And it is time consuming. So we want to automate it using AutoML. We are in the process of doing it.
So after automation, you are able to find the.
Yeah, if we give a query of a function or a gene, there are more than 1,000 molecules that are present in this database. And also, it is not just for cucumber or sorghum. We have more than 50 organisms whose data
Okay, you are.
genomes are there. So it is a vast opportunity for researchers to use this tool and find out specific genes or SNPs and proceed forward.
One last question, like, is there a wet lab validation of this data?
So no, sir.
Okay,
Not-
thanks. Yeah, others can ask. Yeah, I think we can close. Yeah, but next time, please ask questions.
Thank you.
Thank you, participant. The next presenter is J. Akansha on the title GenNOT Pipeline for the Analysis of Plant Genomics Data.
Good afternoon, online present here. I am Akansha JJ from the Department of Plant Molecular Biology and Bioinformatics, following my second MSc in Bioinformatics. I am here to present on GenNOT pipeline for the analysis of plant genomics data. Basically, bioinformatics is an interdisciplinary field that comprises mainly biology, computer science, as well as statistics. So, what is a bioinformatics pipeline? In general, pipeline consists of series of inlets and it will be provided outlet. So, say first, a bioinformatics pipeline also consists of a set of complex algorithms or tools which process the sequence data and produces the output. If the sequence reads is in the form of transcriptomic sequence, differential expression analysis will be done. If it is in the case of genomic read, the annotation and the gene prediction will be done. So, coming to our pipeline, GenNOT is a comprehensive pipeline specifically designed with an application programming interface for the analysis of plant genomics data. So, integrating and analyzing large data sets is very difficult. Even researchers from other disciplines are using bioinformatics analysis to substantiate their wet lab results. This pipeline enables the users to identify their results very accurately. So the methodology or the starting of the pipeline starts with raw reads. Before proceeding with the raw reads, the sample is prepared and the DNA is extracted. Then the library construction is done, QC and quantification is done, along with the sequencing of the reads. Sequencing can be done based on 3 sequencing methods, that is first generation sequencing or a second generation sequencing, which is the next sequencing generation method, or finally the third generation sequencing methods, which is now very famous, that is PacBio and nanopore. Then come the sequencing methods, we'll be getting the raw reads. Those raw reads are then quality checked. If the quality is good, we'll be proceeding with assembly. If it is not good, we'll be proceeding with trimming process. After trimming process, we'll be getting the pre-processed reads. Then assembly is being done. Assembly is also done based on 2 types. If you have the reference genome, we'll be going for reference-based assembly. If you have the de novo, if we don't have any assembled sequences, we'll be going for de novo-based assembly. Based on the assembled sequence, we'll be going for the variant calling files, then along with annotation and the pathway analysis. So the GenArt pipeline consists of a set of tools for bank to SAM conversion, GFF to GTF conversion. In such a case, if a user doesn't have raw reads, instead he's having .sam or .com file, he based on his based on his need, he can convert it. And in the pre-processing steps, we have incorporated FastP, FastQC, and Innovate. And if in the case of de novo, Spades and Abyss is used for short reads, and Hyphism is used for long reads. For mapping of the reads, we'll be using BWA, which is Barrow's Wheels Aligner, and Bauchi. And analysis can be done by BLAST and as well as gene ontology mapping. So the pipeline graphical user interface is done with the help of Python, with the packages Conda and Mamba, with the installation banner of GenArt. It shows the banner GenArt. Along with that, necessary tools can be installed, so that we'll be getting the pipeline GUI, which consists of a set of tools, pre-processing, genomics assembly, analysis, and genomic pipeline. On clicking this genomics pipeline, we'll be getting the window in which we just need to select the forward files, reverse files, adaptive sequence, and the reference genome. By running the pipeline, we'll be getting the output as FastQC output, trimmed output, BWA output, pathway, and BLAST annotation. So, what are the outcomes? So, we are validating with the Proso Millet. In the first step, we'll be finding that the quality of the reads were good. Based on the spread score, if the spread score value is above 30, all the qualities were good. Here we can find the quality of the sequences were good. So we can directly go for assembly. But here I have trimmed the reads, and again after trimming, again the reads were quality checked, and find that all the reads, the spread score of all the sequences were above 30. Then the sequences were aligned using the BWA. And we can find the variant coding files from this section. The sequences that are different in their forms are marked in red color, so that the variant files are found out using the free-based tool. Then the annotation step is done. Annotation is done by using the BLAST to Go, in which we can find the high throughput sequencing, and with the help of gene ontology mapping. Also, we can find the BLAST, which that is the basic local alignment search tool, and BLAST it without hits. So the future perspectives of this tool is enhanced automation, which means like if the user doesn't know where to start, with the help of the automation, if he's having a VCF file, the tool itself finds it's a VCF file, and then the other steps are being done. And AI assistance, we all know these days AI is playing a major role. So as such, by providing an AI, even researchers from other fields can find to start from the previous step, and increased use of cloud computing and distributed systems. Bioinformatics has a huge amount of data, so cloud computing and distributed systems plays a major role, and multi-omics approaches by integrating other omics such as the transcriptomics, metagenomics, metabolomics can be done. and advancements in the real-time sequencing and faster clinical and research application, which means genomics pipelines are playing a major role in cancer research by based on the input fed to the system. So with this, I conclude Zenark is a genomics tool with the application programming interface.
Thanks, sir. It's a good presentation. Is this pipeline freely available for public?
Yeah, it's not yet published yet. After completing, we'll be publishing in GitHub as a free version.
Okay, will you allow any of us to further include or make manipulations in this?
Yeah, sure. Actually, we are having pipelines such as SnakeMap and Galaxy, which are based on command lines, which is not a user-friendly version. And there are commercial versions for also user-friendly like Omixbox and CLC workbench. So this will be a free version and you can be freely accessed.
Any other questions?
Is variant calling has been incorporated in this tool?
Yes, sir. It is done by using free-based tools.
Okay, and annotation for the variants?
Yeah, the blast. I have incorporated blast.
Akansha, can we able to integrate the metabolomics data with the genomics pipeline if possible?
Yeah, by identifying the SNPs, we'll be able to get the metabolites so that we can integrate metabolomics.
Any other questions? I have one question. So in terms of read length and depth, when you compare PacBio and Nano, what is the distinction? And when you combine it in use it newer, what is the final read length and depth that you had?
Sir, actually, it's not like that, sir. By giving the raw reads, even if it's a PacBio or Nanopore, it's not an issue. By giving the reads and reference genome, it will be able to compute all the results.
So in principle, between the PacBio sequencing result and the Nano result, what is the basic difference? What is the basic difference when you get the data from a PacBio machine and a Nano machine? In what terms? Why not only do PacBio? Why you want to have both to have better? Okay, so what is the final read length and depth that your program has used?
Sir, it's not like that, sir. It's based on the software we have already given us backend. So
you don't count that. You don't count that in the pipeline.
Yes, sir.
Okay. So which means that is there a filter for you to have a low depth, low read sequences to filter? Otherwise, you was.
Yes, sir. Based on the FED score, if it is above 30, if it is below 30, we are going for Phenometrics so that it will bring the low quality base pair and it will provide the output based on above 30 FED score, sir.
Okay, thank you.
The next participant is Tessie Sneha on the title Genetic Variability Studies on Yield and Bran oil Characters in Rice for Isa Sativa.
A warm afternoon to all present here. This is Tessie Sneha here before you to present on the topic Genetic Variability Studies on yield and bran oil characters in rice. As we all know, rice is the global grain. As each year increases, there is an increasing demand for rice. But the cultivable lands are getting decreased, but we need to meet out the growing population. But we need to meet out the growing food demand of the growing population. So what can we do? Here comes the role of plant breeders, that is us. We are going to design a successful crop improvement program, keeping certain things in mind. First one is the variability parameters. First one is the variability parameters. We need to choose a plant not only based on its phenotype, because as each year progresses, the performance gets reduced. Hence, we need to choose on the genotype, heritability, and genetic advance. Second one is the correlation. Here, it tells about the importance of individual character towards the yield. It is further divided into direct and indirect effects, like the particular trait directly contributes towards the yield or indirectly contributes towards the yield. Last comes the diversity. No two rice crops are the same. Each one is unique in its own way, and this is what is creating the diversity. Hence, we have to choose diverse parental lines suitable for hybridization programs Thereby, we can get superior recombinants. 2 years before, that is in 2022, we all faced a shortage in the edible oil because of the war in Ukraine. This is because Ukraine is one of the world's largest producer and exporter of sunflower oil. Because of the crisis there, we weren't able to import sufficient quantities of edible oil. So we started to look for the next best alternative. At that time, we focused on the rice bran oil because of its added health benefits. It is because it supported a healthy blood sugar level, it relieved from stress, it had anti-aging properties because of the properties called squalene in it. It maintained a healthy heart, and hence it is rightly called as a heart oil, and it has lots of antioxidants like gamma oryzanol and omega-3 fatty acids, which boost the immunity and its fight against cancer. So, which made us to frame our objective of our research as to evaluate genetic variability, heritability, correlation, genetic diversity among the genotypes for improving the yield along with the nutritional quality. We took a total of 54 genotypes, which comprised 39 land races and 15 varieties. We sown them in the raised beds at the paddy breeding station, and after 28 days, we transplanted them to the main field. And this is the main field view. We took 13 biometrical traits and the variability estimates like PCB, GCB. We used Burton's method, and for heritability, we used less formula. And for the D square, we used Mahalanobis. The software we used was Arthrogram and PNU stat. Moving on to the results and discussions. From the ANOVA table, for the mean gamma squares for all the treatments, for all the traits, we were able to find significant variation, which was found to be good. And moving to the variability table, we were able to find a broadest range for total number of fertile spikelets and the narrowest range for observed for the flag leaf width. And the PCV values were slightly higher than the GCV. It is because of the environment and genotypic effects. And finally, we concluded that flag leaf length, flag leaf width, total number of spikelets, fertile spikelets, test weight, rice bran oil, and single plant yield has the highest value for the mean PCV, GCV, H square, and GAM. And yes, we need to focus on these traits for improvement. Moving on to the correlation analysis. It tells that when we increase the total number of spikelets, total number of tillers, total number of productive tillers, your yield gets increased. But I want to focus on rice bran. Rice bran has got a negative significance, has got a negative association with the flag leaf length. Hence, we need to choose plants with a shorter flag leaf. This is because when the flag leaf length gets increased, the resource allocation for the leaf growth gets increased in comparison with the allocation for the oil percentage. And when you take the other traits, they are non-significant, so increase or decrease will naturally not cause any effect. So this may be due to the environmental influence or maybe the limited sample size. Path analysis, the positive direct effect like total number of spikelets, total number of fertile spikelets has a positive direct effect towards the single plant yield. And rice bran oil has got a negative direct effect towards the single plant yield, and this may be due to some genetic factor. And when speaking about the indirect effect, negative indirect effect, when the rice bran oil percentage increases, the total number of spikelets is getting increased, but ultimately the yield gets decreased. This is because when the rice bran oil percentage increases, though the number of spikelets are getting increased, there may be the production of chalky grains, so which will lead to the reduction in the single plant yield. Moving on to the diversity analysis, the 54 rice genotypes were clustered into 22, where the cluster 3 had the maximum number of genotypes, while the cluster 15 to 22 were monoclusters, telling that they are highly heterogeneous, and hence we can choose these traits for crossing, these genotypes for crossing program. Speaking about the intra-cluster distance, cluster 14 had the maximum, which shows the significant variability within the cluster 14. And speaking about the inter-cluster distance, cluster 2 and 8 had the maximum, which shows the presence of substantial genetic divergence. Speaking about the cluster mean, cluster 3 had the highest cluster mean for all traits except for test weight. Hence, we need to cross the genotypes of this cluster with the superior progeny from other clusters to get the translucent set again. And contribution of traits towards the genetic divergence, rice bran oil, percentage, grain length, and test weight contributed to the maximum while flag leaf with total number of productive tillers and total number of tillers contributed the least. Finally, I conclude by telling that cluster 2, 3, and 8, like core 56, core 23, ADT 58, Chinnar, Arupatam Samba, and Tulasi Vasane Samba, they are highly suitable for crossing in the breeding program for improvising the yield along with the nutrient content, that is the rice bran oil, which I focus here. So,
You said that the correlation between the rice bran oil and the flat leaf length and number of filled grains are negatively correlated, right? So we can increase the rice bran oil content, but reduce the we need to compensate some of the yield. This is optimum for the.
yeah, actually, this is a very nice question. Hence, we need to go for the balanced approach. We need to choose genotypes which are having a moderate rice bran oil percentage, and we need to cross it with the genotypes of the higher yield. When you're focusing on the balanced approach, we can get better.
Did you check the nutritional quality of the grains? So because that is also important. Then if you have high nutritional quality in the bran oil, what about the nutritional quality in the grains which were present in that?
Yeah, very nice question actually. I also check for the macronutrients like nitrogen, phosphorus, potassium. I look for the rice bran oil percentage, and it was like having a very good value.
Any other question? Yeah.
Tessie, how did you sample rice bran oil percentage data? Because what is your strategy? How many volume you took? Because the weight of the brand maybe will be less. How much? How did you sample it? Generated the data.
Yeah, that was a quite difficult time because if you are going to take some 500 grams, you'll get only some 25 grams of rice bran. So what I did was that I collected the rice bran and I kept it in NIR, and I saw for the rice bran oil percentage how much it was, and for oil extraction I went for the soft slit method. And after that I look for the peroxidase value also. So how much of time it is taking for the oil to get transferred. Actually, one thing which I wanted to tell here is that rice bran oil is that much it is not under, it is not commercialized because of the rancidity problem it is facing. Because it has got high amounts of linoleic acid. It is high in polyunsaturated fatty acids. Because of that, your storage, that is the shelf life, is getting reduced. So we need to go for various stabilization techniques. So I think that is one of the reasons why it is not that much in use. But I wanted to tell that in countries like China, Japan, and Korea, they are mainly using this rice bran oil in comparison with India.
Yeah, I have one question you already answered that I was trying to ask too far. So I will go for the next question. Like, you have rice bran oil on one side, maybe a certain percentage of rice bran that accumulates goes for oil production. A larger percentage in the market doesn't go. So because not all rice bran that comes out in the mills is going for oil production. So can we increase that number instead of actually looking for varieties which could, because you're already seeing a negative correlation between yield and the rice bran oil, especially with respect to this question, maybe the nutritional aspects of both grain and the quality of the oil has to be looked at. So is that more towards genetic improvement that you would propose as a conclusion, or instead you fetch all rice bran that are actually otherwise not used and then put it for milling?
Sir, actually, I have referred it. When you go for many article reference, there is no individual variety focusing on rice bran released as a variety. Like this variety has got a largest amount of rice bran oil percentage. No, nothing like that. It is, we are focusing on which lines are having the highest oil percentage. We'll be identifying them to be the donors, and we'll be crossing it with the superior high yielding varieties. We'll be going for crossing program. So while increasing the yield, at the same time, we can provide the nutrient wise also.
So you mean to say the list of parameters to be improved for rice, your rice bran oil quality improvement is one of the last one. I mean, if that is there, you would consider, otherwise you would give importance to other parameters for breeding, right? Is that right?
Sir.
Okay.
Because most in the rice milling industry.
That's good. I mean, you can go for increase the collection and then go for more things. So that's fine. And did you publish this work?
Sir?
Did you publish an article out of this work? Out of your research work?
No, sir, I have just submitted to the journal.
Okay. Okay.
Percentage of oleic acid.
Linoleic acid.
Oleic acid.
Oleic acid. Actually, ma'am, I am not exactly aware of the exact amount of percentage, but rice bran oil has got highest amount of palmitic, oleic, and linoleic acid, which are very good for our health.
Thank you, participant. The next presenter, Mr. A. Surender, on the title Parent Offspring Regression and Multi-Trait Genotype Ideotype Distance Indexing in Intervarietal Cross Derivatives of Black Gram. Vigna Mungo.
Presentation is on parent offspring regression and multi-trait genotype ideotype distance indexing in intervarietal cross derivatives of black gram. So black gram is a highly self-pollinated, short duration and climate resilient crop, which is suitable for various climatic conditions and can be adopted in the various cropping systems and cropping patterns. So since it is having a significance in nutrition, it has a good role in the vegetarian diets. Even though we are releasing the high yielding varieties and we have the more area for cultivation, due to the low agronomic practices and extreme weather conditions and prevailing pest and diseases due to increased weather conditions, the yield potential is the productivity is very low, and the yield loss due to MOME diseases it may vary from 80 to 100% depending upon the weather conditions and the availability of the vectors. So continuous breeding for the development of high yielding and yield climate resilient varieties is needed for the black gram. So due to since it is a highly self-pollinated crop, the genetic diversity is very low when compared to cross-pollinated crop. So the creation of new variability through hybridization and selection by the novel selection approaches will be required. So we made a cross between Co6 and Bamban 11. This was a high yielding cultivar, and having the Co6 is moderately resistant to OMB and Bamban 11 is highly resistant to OMB. These crosses made to develop the high yielding and OMB resistant varieties. These F1 hybrids were confirmed by the presence of hairy pods, which was inherited from the male parent. And in the F4 progeny, the lines are showing almost homozygosity in every line. In the parent offspring regression, we analyzed F3 and F4 generation for all the quantitative traits. In F3 generation, 64 F3 plants were selected based on the yield performance during summer 2023, and these selected 64 plants were raised as the progeny rows in the Kharif 2023, and from these two generations, 9 quantitative traits were evaluated for the parent offspring regression, intergenerational correlation, and narrow sense heritability. In the parent offspring regression, the regression coefficient was highly significant for all the traits studied, and the intergenerational correlation also highly significant for all the traits. It shows that high relationship between the F3 and F4 generations was found. So hence the selection can be affected from the F4 generations onwards. But the narrow sense heritability is high only for the number of seeds per pod. For the remaining traits, the narrow sense heritability was moderate because it may have the non-fixable genetic components like dominance gene action and epistatic components, which was already confirmed through generation in analysis. Hence, the selection at the later generation will be useful for improvement of the trait. And next, after F4 generation, the selection was made through the multi-trait genotype ideotype distance index. This index, these are the advantages of the index. we can plan the video type, which traits has to be increased and which of the traits has to be reduced. And it is free from multicollinearity because all the traits are converted from correlated to uncorrelated factors. And this is having free from weightage coefficient. In order to give weightage coefficient every trait, we focus. And it is easy to interpret. It is simply giving the index based on the low index, that is low distance between the genotype value and the idiotype value. We can select the genotypes. This is a sequence of analysis. 9 quantitative traits were recorded from the F4 progenies of Pro 6 cross Compound 11, and all the traits were rescaled into 0 to 100 values. And these rescaled values are converted into factors to reduce the collinearity. And after that, idiotype planning was done. In this, all the 9 traits were focused to increase at higher levels. So we give a higher 100 score for all the traits, and the computation of MGDA through or package using meta package. In the at 10% selection intensity, we have selected six 4 progenies which are having low MGD index, and these progenies are having they are very close to the desired idiotype. These selected progenies are having higher among the selected among the 64 progenies, and these selected progenies were also having a superiority over the selected progenies. So, analyzing the contribution of the factors, factor 1 is contributed high towards the MGDA of the 53. So, these traits associated traits, number of pods per cluster, number of pods per plant, pod length, number of seeds per pod, and CDE per plant are associated with the factor 1. This factor 1 is highly contributed to the calculation of P53. So, analyzing the broad sense heritability in the F4 generation, all the traits are having more than 90% of the heritability. Hence, the selection will be effective for improving all the traits. Analyzing the selection gain, the number of branches per plant, number of pods per cluster, number of pods per plant, and CDE per plant are having the higher than 20% selection gain. Hence, this selection index can be utilized for selecting simultaneous improvement of the yield contributing traits. So, salient findings of this study are significant regression coefficient and intergenerational correlation for all the traits were found, and high narrow sense heritability for number of seeds per plant, and moderate for remaining traits are found. It shows the high intergeneration relationship among the F3 and F4 generations, and through MGDA at 10% selection intensity, 6 progenies were selected with a high selection gain for yield contributing traits.
Yeah, please prepare a question. Meanwhile, I'll ask one. What is the epigenetic contribution in this conflict? Is there anything known in epigenetics other than the genetics?
Epigenetics, we can't estimate simply through.
Is there a role for epigenetics in the first place in this parental offspring conflict?
I told the epigenetics.
Epigenetics.
Epigenetics is
The one, something like a methylation, few other changes that occurs based on those aspects. Is there a correlation? Like, is there any information in that regard? In the equation. What do you expect? Like, based on, is it purely genetics or?
It's not because environmental factors also influencing in the segregating population. So we can't estimate the epigenetics through these analysis.
Is there somebody done like methylome sequencing?
Yes, sir.
So is there a information out of that methylome sequencing? One for this particular trait? No. Okay. Yeah. Questions? Yeah.
So I could see from your slides that plant type is highly irritable, right? But it is highly environment influenced, right? So we could say it's highly irritable.
through narrow sense heritability, we have confirmed it's having high because we have selected the parents. The male parent is very short and female parent is very high. So the segregating population is almost normally distributed. So in that generation, we found that it is highly heritable. So it may be low heritable in some other populations. So in this population, it's highly heritable.
If there is no more doubts, thank you, participant. The next presenter is R. Jayachandra on the title Enhancing Drought Tolerance Under Field with Osmolytes and Biochar Coating in Blackground.
Good afternoon to one and all gathered here. Because I'm
Slide change panna.
stress-free. Good afternoon to one and all gathered here. Because I'm feeling in front of you like stressful. Okay, I'm Jai Chandra, now pursuing second PhD, Department of Seed Science and Technology. I'm going to present a topic on about enhancing drought tolerance under the field conditions, how the impact of osmolytic chemicals along with biochar affect under the field conditions. Okay. If the black gram survives under the field conditions or under drought stress or not, isn't it or not? Okay, we all know black gram is a pulse crop, most important third most crop. It belongs to the family Fabaceae. Then it also survives under the drought conditions also. I can predict it. So I have chosen up with the variety of the Bamban. 11. Then what are the objectives we now going to see is what are the impacts of the drought tolerance in agriculture, then the role of the osmolytes with biochar, then at last how it affects under the field conditions. So first I have it with several osmolytic solutions with different concentrations under the lab study. Choosing of the chemicals of the osmolytes, mannitol, sorbitol, chitosan, salicylic acid, along with biochar coating. First, I have did priming. We all know in whatever situation also, in under however situation also, if the plant can't survive or it can't, first for all everything we need a basic thing seed only. Because with the help of the seed only, we have moved for everything. Because we want to consume for our seed production and it also for our food consumption also. So for whatever the process, seed is the basic input quality in all our agriculture crop in whatever the field. So in under whatever the situations, which are all the things it can't survive. So we have chosen up with the priming. It can enhance the uniform germination, then faster rate also under its lead up to the good and better best quality in field condition also. So I have choose with different chemicals under the different concentration. From among that, I have did it on the lab study. So I have taken up into the which are the best thing. I have choose gone with the field condition. So first one is the control, then hydro priming. Then along with the mannitol 1%, sorbitol 1%, then chitosan 1%, and then salicylic acid 100 ppm with the biochar coating 100 gram per kg of the seed. So, what are the effects of the osmolytes and foliar application in the field performance under the drought conditions? We go into the see how it can better. So with all those things, I have the essence with and what that impact because whatever everyone is feeling stressed now because we are all doing research work. How you feel I didn't know, but I'm in front of you and our beloved staff. I'm feeling stressed only because at last they will going to ask questions now. If they didn't ask question and okay, but whatever thing under any stress situation also, we want to move upon with at one situation point, whether it is the positive effect or negative effect. So among in the world, everyone having at some circumstance or some situation, we have stress. So how we can overcome? Likewise, the plant also have some less stress. So with the essence of the different chemicals, I have proven the mannitol 1% along with the biochar coating 100 grams per kg of the seeds. It can give the best result in plant quality also, plant height. With the help of that, how I did this means D0, D1, which is nothing. D0 is the irrigator. D1, every time under drought stress condition, we have at vegetative and reproductive stages, we suffer the yield loss up to 20 to 30% in black gram. So, with the D0 is the irrigator, we have given the water fully. D1 under skipping of the irrigation before the flowering stage. 8 days before flowering and after 7 days after flowering. At reproductive stages, at flowering stage, I have skipping of the irrigation and how it yield and how it produce the height I have chosen. So with irrigated also nearby with the skipping of the irrigation also I have got best result only. Mannitol 1%, it have proved the best result in plant height also. Likewise, the flowering along with first days of the flowering and also the 50 days of the flowering also, the mannitol 1% through because the osmolyte in normally at drought condition we have faced more problems in the seed. It can destroy more cellular damages, infection, than everything of the seed. So under use of the osmolytic condition, we can a drought stress condition and all, the chlorine substance which can produce more. So it can enhance and production of the cellular damages and other membrane interjection also. Then the osmolytes also affect under how with the control and mannitol 1% along with biochar coating. I have how it can be essence in field condition. I have given this photo. Bumban level variety in whatever situation also, whatever the farmers want to produce in dry feed areas also okay. Then water irrigated side also okay. If the farmers leak or suffer something means if they put Bumban level variety means they can get better quality as well as also consumer production also best. So among that those things, I have root seed priming with osmolytes, mannitol 1% and biochar coating 100 grams per kg of the seeds. It's very suitable for the variety Bumban levels. If you also try it, we also can better result. Thank you. Thank you.
Actually, some of the treatments are less performing than the control condition. Why it is so?
Because of control normally normal conditions only, but during drought condition, essence of or the flow in mannitol and some enzymes will develop. Endomanus 1,4 like that enzyme and all. It can be translocated to the phloem and without photosynthesis also, if not happen also, these enzymes help and survive better under all. So likewise, some of the chemical also chitosan and chitinase also it can be overact and react. Then I can add it along with biochar also. So bleaching of the nutrients also we can avoid. So uptake of the water is all very essential and we can water uptake and everything we can over.
How could the usage of osmolytes and the biochar affects the seed germination? Since we are doing the priming process, what is its influence on seed germination? We are using control and we have used the combination of the osmolytes along with biochar. What is the influence on the seed germination? Since we are priming it, how it is performing apart from drought, how it is influence on seed germination?
I want to say openly means normally control better only. But normally I say in whatever the treatments we did also normally control is better and best one in all. But in some other situations, if not normally without any treatments only we doing seeds on control. But with the use of the seed priming, we can if not grown also, we can enhance the seed germination with better and high influence rate also, germination rate, then uniformity also equal.
Okay, there is a question.
Is biochar, is it generally getting stick to the seed?
How I did means like biochar. What are the things or how many concentration is the best? We have first choose. Then after that we can normally what we do. If we want to do any chemical means soaking of the chemicals in water only. Likewise, I also soaking the biochar in water. Then like filter it. That way I will coat it. If you can also see means it's coated very nice.
What is the thickness of the biochar over the seed?
It's coated very only.
I'm asking how much thick it will be the seed then after coating.
But it's coated very thick only. But I will see and say on this.
Okay. If generally we know that chitosan is mostly used for drought and all the focus spraying. So if we check these treatments and compare it with general chitosan, which is better? Chitosan plus coating with biochar or coating with chitosan, which is better?
Only chitosan is a chemical very better only, but I did on biochar only. I not did on others. So I think I need it.
Okay, any other questions? I have one. What is the planting density you have? I see in your photographs there are differences in. Is it the regular spacing? What is the spacing that was used in your trust? But in terms of growth, it makes the difference of showing those. Okay. My second question is salicylic acid is usually one of the treatments. How is it influencing your trade when compared to the typical osmolytes, when salicylic acid is not an osmolite? So can you substantiate why you use salicylic acid when your title is osmolite based research and how it does? So there is an improvement. Yeah, salicylic acid வந்து உங்க ட்ரீட்மெண்ட்ல as an ஆர்கலைட் ப்ராஜெக்ட்ல இன்க்ளூட் பண்ணீங்க. அப்படி இன்க்ளூட் பண்ணி இம்ப்ரூவ்மெண்ட் இருந்ததுன்னா என்ன காரணம்? எதனால அது இம்ப்ரூவ்மெண்ட் ஆகுது? சலிசிலிக் ஆசிட் எப்படி?
அது 100 ppm டொமேட்டோ நல்ல பெஸ்டா பெர்ஃபார்ம் பண்ணிச்சு.
100 ppm வந்து drought tolerance இம்போர்ட் பண்ணிச்சா? அது இம்பார்ட் பண்ணிச்சா?
எஸ் சார். முளைச்சிச்சு
எப்படி பண்ணிச்சு?
அது முளைச்சிச்சு சார். பட் மேனிட்டால் அளவுக்கு மத்த இதுல எந்த இதுலயும் எஃபெக்ட் அவளுக்கு வாய்ப்பு சார். ஃபர்ஸ்ட் மேனிட்டால் நல்லா perform பண்ணிச்சு சார். மேனிட்டாலுக்கு அப்புறம் chitosan நல்லா perform பண்ணிச்சு சார்.
Okay, so in the small improvement by 100 concentration, what is the reason? What do you think as a scientist could have done to the plant by the salicylic acid which brought that trade influence? Mechanism and all. Osmolite, I understand why you used salicylic acid. Okay, so thanks.
What is the biochemical mechanism behind the biochar in importing drought tolerance?
Normally it's hygroscopic map, and it also when using it can gain water retention also. Then leaching of the nutrients also with the biochar we can reduce the map. It also helps with the effect along with the osmolytic solution.
So you're not mentioning anything about yield.
I don't know.
Sir?
Yield traits.
Yes, sir. But also.
Then how we can say it is drought tolerant?
Sir, we do sir both in field.
And we said you are.
Seriously, give good result only, sir. But I didn't mention here, sir, yield and all.
And how you can say that we are without irrigation yield the day before flowering? How do you know that the plant is going to flower after this?
Sir, normally I didn't know, sir. Normally how I say me without any equipment or without any unknowledge only I came into PG, sir. So well, under my map guidance only I did, sir. So
Not
we
PhD.
normally under reproductive stage and reproductive stage and flowering stage, we can normally yield is lost, sir. 20 to 30% in every farmers and all suffering. So, okay, we can predict like this critical stages. What the positivity or negativity is going to give. Likewise, we just read, sir.
Any questions from students, right? Okay, thank you.
Thank you.
Thank you, participant. The next presenter, Mr. J. Boniface Anthony, on the title Selection of Superior Rice Genotypes Through Multi-Trait Indices.
Good evening, once again. My name is Boniface Anthony. I am here to present on the topic of the selection of superior rice genotypes through multi-trait indices. So, as you see, the area under the rice production in Tamil Nadu is constant for over a decade. So, it is not significantly increasing or decreasing. But we have to increase our production to meet the growing demand. You can also see the productivity is around 4.4 tons per hectare, and it is lower than other areas of Asia where the productivity is quite high. To meet up, we need to increase our strategies to develop high yielding varieties. So one of the technique is developing a hybrid. So it is proven that around 25 to 30% of the yield increase was already achieved through hybrids, but there are some constraints in the hybrid development in rice. So this is the heterotic magnitude observed in different subspecies. You can see the indica type, and within the japonica type, the heterogeneity is very less compared to the indica and japonica type. But crossing the indica versus japonica again have an issue, cross incompatibility, so that leads to sterility in the F1. To overcome that, the new plant type idea was introduced, that is to develop crosses between tropical japonica as a bridge species, which is quite compatible with the indica type, and developing the diverse parental lines to use for further heterostic breeding. So in this concept, some of the crosses were made earlier with well-known restorer like CB87 or 174 or other restorer, which is already used in Tamil Nadu state, crossed with the tropical japonica lines. Around 250 lines collected from different crosses were evaluated along with the 5 checks for yield performance. So why we need to go for the multi-trait selection in this context is the japonica types are high, the plant height is quite high, and they are quite flowering also, the day is more. And some of the crosses we observe still the sterility is occurring. So apart from the yield, so these characters also considered important for developing hybrids, because short plant life with less number of the layers is preferred, because the female lines which available in Tamil Nadu, which is available in the previous of reduced plant life and less number of the days to flowering. So we need to match it up. So we tried multi-trait indexer. The Smith-Turner index is the old one, which is used earlier for selecting based on multiple traits, but it has some of the issues like multicollinearity, and we need to give the weights, to different traits. That is quite not clear which kind of weight we have to give. Some will give heritability as a weight, some will give genetic correlation as a weight. But these indexes, that MGIDI, FAI, which doesn't require any weightage, and these are also aware of the multicollinearity because it uses the factorial designs. It converts the traits into the factors, but using the same kind of the correlations with aware of the multicollinearity. Also, the GYT biplot that is based on the principal components will give the idea of how each trait is contributed to yield, and this genotype is better for each specific trait yield combination. So total 9 traits were taken. Among that, 3 factors were grouped based on the FDI plus on MGIDI. In factor 1, the major yield contributing traits were there in both the indices, that productive yield as microeffectivity and the seed weight and the plant yield. We given a lesser value, like the minimum value is desirable for the plant height and days to 50% flowering. Rest of the traits we are given the increased value as a desirable. So you can see there's a significant increase in the selection between the original population, that is XO, and the selected population XS. And the selection yield is quite high for the cyclic fertility, which is 11% and 12% and for yield, and 31% for the number of grains per panicle in blood, which is also similarly observed in the MGID. But in MGID, the value is quite higher compared to the blood. So this is the selected lines. The blue dotted are the selected lines, and the line drawn, the red dotted are the non-selected. So we used 10% selection intensity in both the conditions to identify the lines. So this is selected lines in the FAI block. So the strength and weakness view of the MGID give idea that each genotype, how it is associated with the different traits. So we already see the factor 1, which is having a yield contributing traits. The dotted line indicates the idiotype. So the line, each genotype, the line which is each factor is out of the dotted lines are considered as the good for which is nearer to the idiotype of the idiotype. So as you can see, the F1, the factor 1, the genotype 21, 149 and 37 was better. For other traits, that is the plant-related traits, the number of grains per panicle and the plant type, in factor 2, the lines 41, 6, 38 and 118 are best. And for reduced plant number of days to flowering, the 94, 138, 151, 149 and 127 are better. So this is the comparison result from the both data. Among the 26 lines were selected. Among the 26, 24 were grouped combinedly in both the indices, whereas 2 were not separate. So we use the GYT biplot also. So it indicates a specific trait and yield connection. You can see the 110, the genotype is higher in terms of yield as well as the number of grains per panicle, whereas 38 is outperforming for all the traits along with the yield. So this is the selected line. You can see the amylose content also. Some of the lines are the intermediate, and one line is having high amylose content, that is 127, and they all have high yield compared to that of the 2 checks, which is also selected in the indexes CR1009 and 454. So these lines can be further evaluated and used for cropping programs to increase the acres. Thank you. I acknowledge CSIR for providing the fellowship and being the for providing all logistic supports. Thank you.
I will ask a couple of questions. Like, in the table, what mean table? What was the statistical method that you use? Just the mean alone is not adequate for conclusion.
Yes, yes. So those are the lines. We didn't use any statistical method to compare with the checks, but those means were selected based on all the 3 approaches.
And that's fine. Inter-approach comparison finally comes. But still within the table, you want to say anything. So if you have not done so, what would be the possible statistical methods that you could use?
We can do multiple mean comparison tests.
Mean comparison is one, okay.
We can use that.
But you wanted to compare with the check, right?
Yes.
So mean comparison is one. Any other? No? Okay, so to start with, what is the kind of a population or the size of the 30 plus? What was the number that you?
250 likes total.
259.
250.
Okay, what is it? How is it the mini core or where does it come from?
Earlier, previous students, they have selected based on the restorer genes, the RF3 and RF4.
So from what is the bigger population size from which this was narrowed down by your previous research?
The population size exactly I don't know, but from F2 itself, they have followed the marker selection, the RF3 and RF4. Only those lines which just present both the genes are any one of them.
They were actually screened for the same parameters. What you have been screening for?
Earlier, they didn't screen for all the parameters. Only the molecular based selection they have did. the phenotypic evaluation I did, because earlier the segregating
I'm
of
trying to understand how the multi-trait was from the beginning considered while you narrow down here and then why
two traits they have considered, cyclic fertility along with the marker, because that is the major issue they have faced, and plant height, the reduced plant height, because all the japonica lines are exceeding 130 centimeters. So they considered two traits along with the marker evaluation, they selected, then they narrowed down.
And of these ones, how many of them are on par with the regular cultivated varieties in terms of its performance?
Some of the lines were around 30 lines. I have again evaluating the multi locations, which are almost on par with the 454, the variety and CR 1009. Still, those results I didn't analyze. I have to check how it is performing better in all the different environmental conditions.
Okay. What is your future plan now?
Future planning this aspect.
Yeah, how to take this forward now?
So, my major work is to identify the amylose.
In your work, I think you completed already. Okay. But what would be the scope of this project next?
Some of the lines they are already using for the hybrid development. 2 line and 3 line, both the system they are in the process. And based on earlier finding one of my colleague, she finds some higher heterosis compared to indica type. So they are planning to go for advancing these lines in developing hybrids, both in case of 2 line and 3 line.
Okay, I'll stop here. Any other questions? Students? Okay, if not, thank you.
Respected dignitaries, we just have a 2 minutes break to have a technical issue to sort out it.
Another present here. Today, I'm going to present in the topic pre-dollarized therapy, advanced analysis of solving free volatile organic compounds and statistical revelations. Seed deterioration is an irreversible, degenerative, and inexorable process which decreases the ability of the seed to survive. Usually, oil seeds deteriorate faster than any other crop. It is because of the high oil fraction, absorb more moisture, lead to self-heating and seed damage. Oil seeds usually having a more lipid peroxidation, producing more fatty acids, leads in increased cell acidity, which in turn detrimental for the normal cellular metabolism. The crop which I have, oil seed crop which I have been selected for my study is on. The oilseed crop which I have been selected for my study is soybean. Soybean dominated the oilseed industry with 59% of the total global oilseed production, in which Brazil stands first with 40% of the total production, followed by USA, Argentina, China, and India and Paraguay is in the fifth position with 3% of the total production. Soybean known for its high protein and oil content. This 15 to 25% high oil content is composed of 51% linoleic acid, 10% alpha linoleic acid, 23% oleic acid, 10% palmitic acid, and 4% stearic acid. These polyunsaturated fatty acids, when undergo oxidation, produces a free radical. These free radicals are nothing but an atom or molecule with an unpaired electron. Once the free radicals are produced, they continue to propagate furthermore free radicals. These free radicals then converted into hydroperoxides. Hydroperoxides are nothing but the primary oxidized products. These primary oxidized products then decomposes to produce a secondary oxidized products, which are called as volatile organic compounds. These volatile organic compounds are low molecular weight components with high vapor pressure, and it includes diverse chemical components like alcohol, aldehyde, acids. Usually, very subtle variation in the seed quality is very subtle for traditional evaluation techniques to catch up. So, this study aims to explore how the volatile organic compound analysis could be used better to understand the seed deterioration of soybean seeds. Here, freshly harvested genetically cleared soybean variety has been procured and kept in an airtight container, modified with an septum for a collection of gas samples and stored in an ambient condition for a period of 8 months. Then the gas samples were collected within solid phase micro extraction fiber and subjected to GCMS analysis for volatile profiling. Meanwhile, the stored seeds were subjected to germination test. Here, volatile profiling revealed that a total of 76 different volatile organic compounds were released, which include 10 alcohol, 19 aldehyde, 15 acids, 6 ester components, 12 alkene, 4 alkene, 8 ketone, and 2 amides. Meanwhile, the stored seeds were subjected to germination test, and the germination report shows that the seeds were decreased in germination from 76% to 59% over the period of 8 months. To identify which are the signature volatile compound responsible for the seed deterioration, we conducted a correlation analysis between different volatile organic groups and the germination. Here in this correlation matrix, blue color indicates a negative correlation and the red color indicates a positive correlation. And the size of the circle, lower the size of the circle, higher will be the correlation. And in alcohol components, ethanol and tetrahydrochromanol have a highly significant negative correlation. In aldehyde, many of the components are having a highly significant negative correlation. Coming to acids, benzene dicarboxylic acid, octadecanoic acid, and acetic acid are having a highly significant negative correlation. And in case of an alkane, cyclooctasidoxis and pentoxocyclopentad again having a highly significant negative correlation with germination. And then remaining groups, this hexyl phthalate is having highly significant negative correlation. Overall, in these 76 volatile organic compounds, 15 were identified as a highly significant negative correlation with the germination. Then coming for principal component analysis, this pie plot shows that principal component 1 and 2 are having a highest variability of 67% and 26.8%, with an eigenvalue of 6.02 and 2.41. These principal components, along with the factor loading, shows that they are volatile components, acetaldehyde, ethanol were highly associated with principal component 1 and hexanol was highly associated with principal component 2. These three volatile organic compounds were identified as the signature volatile compounds responsible for the deterioration of soybean seeds. These key findings highlight the vital role of volatile organic compounds in seed deterioration. So it can be used as a potential biomarker for using these volatile organic compound to assess the seed quality in a future seed quality control program. Thank you.
Yeah, questions. How much time did she take?
5 minutes, 10 seconds.
5 minutes. Questions? Yeah, go ahead. Yeah, go and ask question.
Very good presentation. And can I know about what is the variety used and what's the moisture percentage of the seed when it is being subjected to storage, and what's the temperature of storage and the lighting will be maintained in the conditions where it is storage, and also what's the material used for storage of the seed?
Okay, the variety is JS335 and the moisture content is 8%. It is stored in the ambient condition, that is room temperature. It is just stored in a storage room. And the other question is
Lighting humidity and temperature, lighting humidity of the
25 degrees Celsius.
RH percentage.
Sorry, temperature is 25 degrees Celsius and RH is nearly, I think 90. Sorry.
Can you just explain the sample preparation for the GCMS? Since we are storing at different months, are you using the seed sample directly or how did you extract the sample?
He treated the volatile organic compound.
Only the gas has been included, but what is your sampling strategy followed?
I think volatile emitted by the seeds are collected using solid phase micro extraction fiber, and it is directly subjected to decentralized analysis.
And in the heat map you have displayed, there is a variation between the 5th month of the storage and the 7th month of the storage. Certain compounds are hiking the month of the storage, the heat map analysis which you have presented. What could be your inference? In the median time of the 4th month, 5th month, we could able to see a range of compounds, whereas in the final 8 months after storage, we couldn't be able to see any alcohols, aldehydes. What is your interpretation on that?
Some of the solvents or acids.
Thanks.
Any other questions? Yeah, just a follow-up of this question, in that methodology, how do you make sure that the control, like do you keep some scrubber or something to remove completely so that when you run the sample versus the one that to remove, so what is the control that you used in the process? Because it's a volatile, right? Like, is there a way that you can completely remove everything and you have that captured in the GC that would not show all these compounds? Is there any universal scrubber that you used? So how did you compare? You compared with what? Like what was your blank run in your GC? So all are samples. Okay, so then how would you make sure that there is no, I mean, is there a standard like positives that are positive, like you have a mix of gases that has representation of, let's say, 10 compounds that you would otherwise estimate so that you can compare? So how would you explain your results in the absence of neither negative control nor a positive control? All that what you captured is that you can compare with. You get my point. But what is, first of all, what is the control used in this study? Is there any control that for this experiment you use?
Sir, can I substantiate?
Yeah.
Since we have done the heat map analysis at the initial stage, this could be used as a control. So that on the day without storage can be used as a control, and the increase in the volatile nature can be taken as a or.
So in that case, how would you uniformly keep the conditions to make sure that the representative, let's say you are profiling this time, and it gives a profile of, let's say, 70 plus compounds. And after 10 days, using the same setup, I'm not sure maybe whether it can be related to number of seeds that you use, the volume of that plant that you handle. There is the normalization that comes in that allows me to, either if I repeat it next time, it would pop up the same one in terms of number of compounds that it reflects as well as the quantum on which that it reflects. So I see that there is a bar that you can establish by taking an initial reading and then comparing it with the other one. That's good. But still, I would expect to have, otherwise, every time when you repeat it, you get a different result. You use normal jars of certain size, certain seed numbers, certain, so where is the normalization comes in all your treatments and number of times that you do? Is there an explanation for that? Okay. And how this volatile profile can be related to food? I mean, soybean as a food, as a quality. Is there any reflection on that?
The seeds,
Certain composition, seeds of variety from some composition can have a different palatability as a food, as a when you process. Is there a correlation between food quality and the volatile composition? Do you expect one? It's a guess. You guess that there will be a correlation. Is there a correlation between, let's say, a storage pest versus this composition analysis? Storage pests and the composition's influence on occurrence or non-occurrence, type of pest that comes.
influence on volatile organic compounds.
So in that case, can you tell what are the key compounds that are in this list would be responsible for either reducing or increasing or attracting the pests? What is the relevance of your study? Can you articulate the relevance of these findings in a practical scenario in 1 or 2 lines?
These major volatile compounds can be used for seed quality analysis. We made a sensor prototype like sensor means while going for storage, we can sense if the seeds start to release these signature volatile compounds. We came to conclusion that it starts deteriorating. Started deteriorating.
Seed quality and storage duration. That's the only thing that you would project as a relevance of this study, right? Okay. Okay, thanks.
Sir, can be able, a study can be able to go like a standard sowing and harvest the fresh seed and can use it as a control. Is that any one of the options? Or secondly, can we store the her genotypes permanently at -80 or -20 and every time when she does DC sampling, if she would have sampled.
Yeah, no, actually
So
there
it
are
could be-
more to discuss here. Even, I mean, let's say how, let's say the fees normally at these conditions when they analyze maybe 12%, 10% moisture or something. So that would amount to create lot of changes in the respiration and its storage extension. So that should be the primary control. Second one, third, there must be a chemical component that is actually used as a standard to run while running the seed itself. So first itself, we have not seen the chromatogram, we have not seen any of the just final results we have seen. So how would you actually even, you are getting some peaks and you're basing it library and then listing the compound. There's no control from any angle that I could see, or at a reference point, or it's a uniform start material. So those were all the, yes, that could be right, what you're saying. But there is more to explore here to refine it. But many can, once the volatile comes, the technique itself is challenging. And the process from which you would start the sample should be as much carbonized as much possible so that the volatile compound, otherwise it will show some peaks, but yeah. Okay, thanks. Yeah, we can
Thank you,
discuss
sir.
it.
The next participant, Mr. K. Gowda, on the topic establishing lethal dose LD50 thresholds for the induced mutational drives using gamma rays and ethyl methyl.
Good evening to all present here. I'm here to present about the topic establishing a lethal dose LD50 thresholds for inducing mutation in rice using the gamma and ethyl methyl sulfonate. We know that rice is one of the most important dietary cereal crop which nourishes major share of the world's population. Even though rice has a major share, but now we are facing climate change and various factors which reduces the production. For that, we need a more improvement techniques, more enhanced breed techniques to improve the crop production. Here we use a mutation breeding technique to enhance the existing potential variety. We know that rice variety AGSC, which is released from rice research station Ambasandram, has the pedigree of ADT 31439 released during 1986, which is the major cultivated variety of southern districts such as Tirunelveli, Thoothukudi, and Kanyakumari, carrying the 90% over past 30 years. Even though it has a good suitability and potential, it has grain shattering and pest and disease susceptibility problems. Mutation breeding is a more suitable breeding technique for the self-pollinated crops rather than the cross-pollinated crops due to that high suitability and require few generations to fix their trait in the mutants. We know that during 1950s and 60s, there are the big famine. The mutation in the spontaneous mutation in a gene known as Orisa sativa gibberellic acid 20OX, commonly known as DESD1 gene. The spontaneous mutation gave the green revolution. So such that after the mutation importance is known, the induced mutagenesis have grown. The International Atomic Energy Authority, Austria, they developed, they are the nodal agency for mutation breeding. Till now, around globally 3,662 varieties were released over 240 crops in 75 countries. Among that, 836 rice mutants were released using physical and chemical mutagens. We use induced mutagenesis will be fruitful for the genetic calculation for enabling the ASD16 variety with the desired trait of interest with the genetic background of ASD16. Our objective is prior going for the mutation breeding. The preliminary step is to pick the LD50, that is lethal dose, which gives the mortality to the 50% of the population, which is treated with the mutagen, either physical or chemical mutation. Now, why we take that lethal dose 50 means the mutation frequency after causing the mortality of the 50%, the mutations, the changes created by that dose will be maybe stable and it will be transferred. The dose is far away from this LD50, they are not maybe much stable in the upcoming generation. That's why the objectives are to fix the LD50 of gamma rays and EMS to generate the Newton lines in rice variety LD16 under the in vitro and in vivo conditions. Then to study the M1 generation of LD16 variety treated with gamma rays and EMS for seedling trials. Our selected material is for we selected a physical mutagen as gamma and chemical mutagen as a ethyl sulfonate. Why we selected gamma and EMS is out of the globally released rice mutants, these out of physical mutagens really used, about 80% of the mutants developed based on treated with gamma rays. And EMS also out of the chemical treatment, EMS is most probably used, and the treatment procedure is quite simpler in the EMS. The gamma rays, the plant material we taken is dried, well-filtered dried nucleus seeds are taken from rice station Ambasamudra, and they are treated in the Nama Chamber installed at the Center for Plant Building Genetics in TNA. And chemical treatment was done for in the Kiligulam campus. Our treatment dosages along with control, the gamma, we take 100 to 500 days, and in EMS, we take 100 millimoles, 125, 150, 170, and 200 as a treatment combination. For the estimation of LD50, estimation of LD50, we used probit analysis. It is a inverse cumulative distribution function or a quartile function associated with the standard normal distribution. In this, the number of mortality occurred in the mutant population, mutant seeds and the control seeds were compared, and the probit values will be calculated based on that the LD50 will be fixed. So the probit analysis values in vitro, the seeds were treated with the gamma and EMS were grown in the generation paper in the in vitro condition, and as well as in the raised bed in the field condition. In vitro, gamma rays, as the germination percentage mortality were calculated, the LD50 for this about is 345 grade around. For EMS, the in vitro value is 133 millimoles. For in vivo field condition, the gamma ray LD50 value is about 360. And for in vivo, EMS is 140. The variation between the in vivo and in vitro is due to the environmental information. This is the graph for LD50 fixation of gamma rays treatments for probit analysis. And this is for the EMS treatments. This is the shoot and root length variation among the gamma rays and EMS treatment observed in vitro. Along with control, the increase in the treatment dosages, the shoot and root lengths are affected greatly. This is the in vivo observation. These are all the data obtained such as germination percentage, root length, and pollen fertility percentage. They are significantly reducing as increasing as dosages. This is figure 3. This is a pollen fertility study compared to control. The dosage increases, the number of sterile pollen grains getting increased in both gamma and EMS. The deliverables are LD50 dose for gamma rays under in vivo and in vitro conditions are respectively 345 grays and 316 grays. The LD50 dose for EMS is 133 and 143 grays. The future thrust is the LD50 value assessed by AC16 would be helpful for the genetic enhancement of the variety from mutation breeding. The preferred desired lines will be fixed by the selection from mutation generations, the advanced, and retaining the other features of AC16. Thank you.
Yeah, go ahead.
What is the distance between the source cobalt-60 and the seed treatment? There is a distance right from the source. When we are treating with gamma radiation in the gamma chamber, there will be a distance between the source, this is the center, cobalt-60, and the seed which you are placing. So what's the distance which is maintained? Because it's also important for thinking.
Yeah, but I gave the seed samples to a gamma chamber, and the assistant will be treated. I won't much advance.
Any other questions?
Is this mutational breeding still a prominent one as we have many other advanced techniques?
Yeah, we have many advanced techniques. Mutation breeding also, we have advanced this random mutation what I did. We have advanced gene editing techniques like CRISPR, and all that now are emerging advanced. But these are already existing one and promising. They proved they proved and provided already 856 mutants all over globally. That's why we tried.
I have a question. See, in the literature, is this information not known because the LD50 for rice, I'm sure this both gamma and EMS based mutations are there for decades, maybe 80, 90 years, maybe 100 years. So what is the importance of this study that you have to establish this LD50 dose now?
The LD50 dose for this variety we can fix. Then the
varietal dependent, I thought it's in general for rice.
This is the varietal treatment. The LD50 values will vary depending upon the seed material what we use and the moisture content and source we use and the duration of the treatment based on the LD50 varies.
Okay, and I'm sure it will vary, but it could be a very narrow range, right? Is there a need for a separate thesis on that? I mean, how would you substantiate your work? The need for this result, or how would the results that you already have since you completed it?
Based on these results only, I raised based on this I will confirm the LD50 and the treatment dosage above the LD50 and below the LD50 that treatmented mutant populations only I will raise the amount and
I understand that point. You have standardized it for your study, that's good. But compared to literature, that so many people in the literature reported for rice, this is the range of LD50, and gamma radiation, this is the range in which they can. So how would you compare with that? Is there some difference that you found that the previous ones are much improved out of your results? Like, is there
not that much?
So what is the significance of your study? Because your title itself is to it's kind of a methodology establishing the LD50. That's the prime agenda, right? So how would you substantiate your results in the current perspective, 2024? I'm sure you can do, yeah, go ahead. Then. Just compare it with the literature and say what's new that you found or what clarifies that.
The older literatures, they did the LD50 values and based on the results, they go for the further analysis. That I followed the same and apart from that, biometric study and analysis will be.
Okay. All right.
Whether mutation efficiency or effectiveness vary with genotypes?
It varies with the dosages. Genotypes, it may be vary, but not that much. The variations will not be there.
That's first.
Effectiveness is not that much really. The effectiveness will be we can calculate only after M2 generation. Thank you.
Thank you, participant. The next presenter, M. Pavitra, on the title Synthesis and Characterization of Versatile Integrated Organic Polymer for Sustainable Crop Production.
Good evening to one and all present here. Myself, M. Pavitra, third PhD from the Department of Seed Science Technology. I am here to present my topic on optimization and characterization of quercetin quantified through HPLC analysis for improving seedling vigor of green gram under abiotic stress condition. So the crop which I have chosen is on green gram. We all know that pulses is the second most important crop when it comes for human dietary consumption. And especially this green gram is the richest form of antioxidant. But when it comes for crop production, the crop production is affected due to climate change. And based on statistical data, 84% of the yield loss is due to climate change, and among that 84%, 86% of the yield loss is due to drought stress. So to mitigate this drought stress and also to improve the seed production, there are several seed management strategies. So among the different seed management strategies, pre-seed integration technology is one of the most important technologies in order to improve the seed production, also to increase the seed, even under abiotic stress condition. So nowadays, everyone is focusing on the eco-friendly farming. So that's why we have chosen a plant-based product, like application of plant-based product at optimum time and at optimum quantity will increase the seed production from 82% to 90%. That's why the gomolecules which are rich in quercetin and flavinoid has been extracted through extraction method and it has been quantified through HPLC analysis. So the first part is on materials and methods. The fresh leaves of guava were harvested from the Department of Medicine and Aromatic Plants, and after which the leaves are dried for a period of 5 days under shade, and it has been dried under sun for a period of 3 days. After which the seed has been powdered under hammer mill unit, and finally the fine powder of 0.2 mm size were obtained. So in order to extract the active component, that is the flavonoid in an optimal concentration, the efficiency of extract is also influenced by various extraction methods. That's why we have adapted two different extraction methods. One is decantation method and the other is soft-select method of extraction. In decantation method, 1 gram of powdered guava seed has been soaked for in 10 ml of water. Likewise, 2%, 3% at 3 different concentration of guava leaf has been soaked in distilled water for a period of 24 hours, after which the leaves has been filtered through Wattman number 1 filter paper. But in case of soft solid extraction, again 5 gram of powdered leaf has been transferred to a thimble and it has been inoculated with petroleum ether, and the soft solid has been run for a period of 8 hours by 6 to 8 siphoning, after which the extract has been diluted with hexane since it is of non-polar in nature. Finally, the seeds of green gram has been soaked into the extract for a period of 4 hours at 3:1 ratio of C to solution ratio, and after which the germination test was performed under temperature of 25 degrees Celsius at 90% of relative humidity. And 8th day of germination, the physiological parameters like germination, root length, shoot length, dry matter production, and bigger index was noted. Where among this physiological trial, we could able to see that maximum germination percent and bigger index was noted in soft solid extracted method, that too at 1% of concentration. This may be due to the fact that the presence of quercetin flavinoid has involved in the mobilization of the enzyme responsible for germination. That's why it has enhanced the activity of amylase protease and lipid content. That's why it has improved the germination. But whereas in decantation method, we could be able to see the decreased germination of 66% and reduced bigger index of 2046. This may be due to the fact that the extract may be of crude form. Since it is a crude form, there may be several inhibitors that would have inhibited the germination. That is, the available form of active ingredient has been inhibited by several inhibitors. That's why it has been reduced the germination. So this is just a hypothesis that the quercetin may improve the germination. To confirm the presence of this quercetin, again the HPLC analysis was done. For that, the 0.5 gram of green gram seeds were soaked in HPLC grade methanol, after which it has been macerated with macerated. Again, it has been centrifuged at 10,000 RPM for 20 minutes. Then the filtrate was injected under optimum chromatographic conditions, and we were with the optimum chromatograph that the presence of quercetin was detected with the peak area of 0.025 under the retention factor of 0.435. So from this, we could able to confirm that the presence of quercetin, that is the flavonoid, has influenced the germination percentage as well as the physiological parameter. Also, there is one more fact that this quercetin has a potential to cope up with the crop to overcome the moisture stress condition. Like it will interfere into the root architecture and it will make the availability of several nutrients and water for the seeds even under stress condition. That's why the seeds of as we have detected, this guava leaves are rich in quercetin content. The commercial form of quercetin is being procured and it has been treated with a seed of green gram at 1% concentration. after which the seeds of quercetin was exposed to polyethylene glycol. Since we have chosen this polyethylene glycol, it has osmotic pressure, it will induce osmotic potential. That's why the plant will undergo stress. So to fix the optimal concentration of polyethylene glycol, again, different concentration right from minus 0.2 megapascal to 1 megapascal has been used, where in that maximum germination was observed when compared to other stress condition like minus 0.4 to 1. 62% of germination was observed in pegging used condition. This may be due to the fact that this 1% quercetin has modulated the enzyme which are responsible for stress condition. Also, the other parameters like peroxidase, superoxidase mutase were noted. Even this 1% quercetin has reduced the availability of lipid peroxidase, like it has reduced the availability of oxygen and it has increased the lifespan of the cities. So the future trend or future threat area is, as I have said earlier, pre-sowing seeds integration is one of the most important technology. After standardizing this quercetin concentration, we can able to infuse this quercetin to any of the organic polymer so that we can able to coat the seed for increasing the germination for sustainable agricultural seed production as an eco-friendly approach. Thank you.
Evening. Since I'm working in flavonoids for the pest resistance, how could you say that the compound isolated has been so stable? Because quercetin is a type of compound which is so unstable, and then you after the isolation condition, how did you maintain that?
Actually, I have confirmed whether the quercetin is present or not. After confirmation, I have purchased the purest form of quercetin. Only with that quercetin, I was doing my research, not with that extract. Just to confirm the presence of that quercetin, I have done.
So what was the extract's total composition? Like, at least the major ones, like, I mean, as I was expecting, as you were asking, similarly, I was expecting you got some profile and from there you somehow came to know that quercetin is playing a role and then you took up your research based on that. But it looks like you have two different things. So what was the composition of that? What are the major Goa extract in the means?
Based on literature, the active compound quercetin, I have.
Did you subject it to the metabolic analysis, the
So
extract?
Theme1 analysis also we have did, sir. In that also the presence of quercetin is being confirmed. But I have done it.
Quercetin is there, but other than that, what else was?
Several alkaloids like hexane, polar, several components was there.
So is any of them not to play a similar role like quercetin? That extract is something useful.
Is quercetin is the major component.
Okay, and you have the data? I didn't see the data. Did you? Yeah, the total composition did talk about it.
Total components, sir, but only the quercetin pKa percentage I know.
Okay, so you are not sure about, you just know that there is a predominant amount of quercetin present in the extract. You don't know about the remaining part.
No, sir, I didn't present it here, but.
You have the data?
Yes.
Okay, yeah. And what is the actual role of this quercetin in your trait improvement? Like, what does it do? Like, because quercetin is also known to do other things, like namely biotics, this related, you know.
It's only to overcome the moisture stress I have done.
How does it do?
So it will modulate the root architecture. So also it will interact with the plant hormones like auxin, cytokinin. It will.
So when you say one chemical is interacting with other, how does it do? Like, is it like as IAA or acetic acid interact directly with the chemical of quercetin, or is it playing a role in inducing or changing the pathway?
Not the pathway.
What do you mean by quercetin interacting with AG, auxin or hormones?
So just by inducing cell division and elongations, it will help in overcome this stress.
Okay, yeah. Any other questions?
Quercetin. What is the cost of this quercetin in pure form and how it will be economical to the farmers?
Actually, it is somewhat costly, ma'am. We are not going to use it as it is, ma'am. By mixing or integrating with some alternative polymer as a base, we will be using. At that time, the cost will be minimized, ma'am.
Yeah, the possibility of doing with the nano formulations.
Yes, ma'am, it is possible to do further.
I have a doubt. As Brad have mentioned about the composition of the metabolites, it's not what on GC analysis we could able to get 50 to 60 compounds in our extract. Have you analyzed your GC profile after your treatments?
Like after treating.
Yeah.
Yes,
Have you
after.
analyzed the GC
After
profile?
treating.
Okay, then what is the nature of your quercetin prior and after at the raw extract solution?
Actually, previously it was, but both case also the quercetin has been detected.
Okay, fine. It's about detection. Okay, but there may be an interplay with all other metabolites present in the extract so that it will give a combined effect. Since the compound is very unstable, can't able to make it store the form, but it cannot able to be, as you have said, we can able to commercialize it. There may be issues with their compound stability, right? How can you?
That's why we have to procure purest form, not that crudest form.
Okay, fine. Even the purest form on deduction of in our GC itself, we must need some certain temperatures and certain concentrations. So when if you are going at as ma'am have said about the field level applications, how could it be useful? Because like we need some particular standard in temperature, pressure, and particular concentration. Only the quercetin is stable, even in GC profile and LC profile. Since I am working with that, I have a doubt. Can say that.
No, actually the presence has been detected. We can't use that extract as it is. Okay.
Thank you. And the final presentation, Divya on the title De Novo Transcriptome Assembly and Analysis of the Genes in Different Tissues of the Background.
Good evening, everyone. Myself Divya. Currently, I am pursuing my master's in bioinformatics. I am going to present on de novo transcriptome assembly and analysis of genes in different tissues of black gram. We all know black gram is also called as woodbine dal. It is a highly important legume crop. It is holding significantly in traditional, nutritional, and also cultural practices. Importantly in South Asia, the genome size of black gram is 574 megabase per. It is a self-pollinated diploid crop. We all know transcriptome information of anything. It will be useful for analyzing the gene expression and also identifying the transcription factors and also non-coding RNA, and also it will be useful for developing the molecular markers. Taking all the advances of genome resources, my objective of my study is to develop the transcriptome data sets for different black gram tissues and also de novo assembly of those different tissues. For that, we choose the Indian popular black gram variety Ramban. The duration of this variety is 65 to 70 days. It is highly resistant to biotic stress, mainly mung-bean yellow mosaic virus and woodbine leaf twinkle virus. This variety contains the totally 21.99 percentage and 77.5 arabinos. It is suitable for growing in all seasons. Coming to the methodology, first we raise the crop in greenhouse condition. After 2 months, we collected the different tissues, the roots, leaves, parts, and seed. Then we did RNA isolation, followed by library preparation and RNA sequencing using Illumina NovaSeq platform. Then we got RNA sequencing. Then we did bioinformatics part. First one is quality checks and RNA processing using FastQC and Gemomatic and FastV software. Then we did de novo assembly using Trinity software. Then for identifying the unigenes, we used CDKit software. Finally, we did annotation using Omics Block software. Coming to the result, this is the summary of raw and clean reads of different tissues. Actually, we got more than 30 million reads for all different tissues. The sample read length is 159 base pair. The GC content is varying between the 44.7 to 48.3. The Q20 value is 97%. The Q30 value is for leaf, it is 93%. For part, 93%. Leaf, 98%. For root, 94%. The table 2 representing the difference between the transcript and gene distribution. Totally we got 2,75,860 transcript, unigenes 2,55,934 unigenes. The maximum length and minimum length is same for transcript and unigenes. The average length for transcript is 825 base pair. For unigenes 784. The counting N90 value is for transcript 293, for unigenes 286. For functional annotation, we used actually for 6 databases, namely NR and NP, Uniprot, KIT and KOG database, and GeoAnnotator. Among these, the NR database gave the high percentage of annotation, 61%, for NP 54%, Uniprot 50%. KIT, only 3% we got pathway result. Then KOG is 56%, the GeoAnnotator is 54%. This figure 1 represents the RNA amplification of different blackgram tissues, seed, leaf, pod, and root. This figure 2, the distribution of unigene and transcript length. Mostly the unigene and transcript length is between 300 to 500 base pair. The 3000 transcript is having greater than 2000 base pair. Coming to the gene ontology classification, in biological process, mostly cellular process and biological regulation and also reproductive process are playing major role. In molecular function, the catalytic activity, transporter activity, transcription and translation are playing main role. This is the top 20 KIT pathways. First one is purine metabolism having 1000 to 19 genes. Thymine metabolism is 1149 genes. This is the distribution statistics of the BLAST result. First one is similarity distribution. Totally 42% is having 65 to 80% similarity. The 24% is having 80 to 95% similarity. This be showing the e-value, the BLAST result e-value. In KOG classification, the orthodox groups mostly cytochrome P450 family and repeated proteins are also involving, and protein kinase family is also involving. In identification of transcription factor, the WT40, like it is highly present in all the different tissues, where it is playing major role in signal transduction and transcription translation activity, and also plant development process. Deliverables. This transcriptome dataset mainly useful for the genomic resources of blackgram. This will be helpful for understanding the genetic makeup of this crop, and also it will be helpful for identifying the important pathways in this blackgram, and also gene families, and also what are the domains are involved in this blackgram. In future, we are going to develop the molecular markers such as SSR and SLV. It will be validated using different Vigna species. Thank you.
Yeah, questions? Yeah.
Nice presentation. Before you are showing some data, shows Q20 and Q30. What do you mean by Q20 and Q30?
That is the quality score. It's like credit score valid. It is showing quality of the raw mix, Q20 value and Q30. It's a some parameter.
Thank you.
Yeah.
Is there any reference transcriptome sequence?
Yeah.
Any other question?
Normal conditions are there any differences for the different?
Normal conditions.
What are the different tissues you have taken?
seed, leaf, root, body.
Where is that final outcome of pathway differences that we found across the different tissues? Is there a final outcome that we found? Like, let's say you take an example of one or two pathways and how it differs across the tissues.
Yes.
So what is the final conclusion you would like to say out of this comparative analysis? Because it's supposed to conclude something, right? So what is can you name something based on your experience that so and so pathway has been found in one, found in, not found in another? Unique to one particular organ and not to another tissue?
In root, there is more number of genes compared with
That would be a little vague statement, but I'm just trying to understand naming a pathway, naming a particular class of genes or group of genes, present, not present, or abundant, not abundant, highly expressed, low expressed, generally across tissues that you studied. Final outcome would be something like that would be good, right? Yeah. So, but even though you have not shown it in the figures, but by experience, you might have gone through all the results that you talked of. Did you find anything that is unique, that so and so pathway has been unique, like let's say a particular amino acid biosynthesis or alkali biosynthesis or anything that is unique?
No, sir, we identify only transcription factors and paper.
So even in the transcription factors, do you see something not expressed in root but expressed in aerial parts?
Yes.
Is it known in Arabidopsis that such things exist? Arabidopsis is one of the well annotated things, right? If you go to the EFI browser, you can find it. And you are able to get the information from the Arabidopsis and then use your data to see whether that is also seen here in background. So according to you, what is the next future application or plan for this final results that you got?
Sir, we are going to develop molecular markers.
Marker is one direction you already mentioned. Yeah, but as a group of analysis, because this is being done, blackgram is less, comparatively less
Genomic.
So how would you use your results to pop up next future plan for either you or for somebody else?
Identify what are the genes are present in different tissues.
Okay. Okay. If there are no questions, we can close with this. You want to say that?
So we have come to the end of this session. One oral presentation. We had 11 presentations, starting from DVO or transcriptome analysis, then fixing LD50, genetic variability studies, genome pipeline for the analysis of plant genomics data, and also there are 2 presentations throughout. And I congratulate each and every one of you. Everyone has presented well, except for the time factor. Right? So, of course, we told you that it would be 10 minutes. You might have prepared for 10 minutes, but suddenly, due to paucity of time, we have to restrict it to 7 minutes. This is one which, and you have to take care of what Sir has informed, because he has given good comments. So, before you submit your thesis, try to include that in your thesis, which will add value to the work. Those who have completed, of course, you can't include it now. Anyway, now I will leave to the chairman, Dr. Senthil Kumar Muthappa, for his comments.
Actually, I have only one statement to make. Excellent presentations, all of you. I was discussing with the professor also to see and also madam that, okay, how many awards are totally there, so that we can try to see, because everybody has done well, unique well. So somebody getting rank of 1 or 2 does not mean that others has a different thing. So we have a very tight competition, and everybody's work is good. Everybody has put in an effort to make sure that the slides are well prepared, organized, and both those who are actually completed and then submitted thesis, as well as those who are going to submit thesis, have put a lot of Yeah, so I congratulate each and every one of you who has come forward to present, because this has already undergone one set of preliminary screening by a committee, and which again indicates that your performance was good. That's why you came for oral presentation here. So I wouldn't give a lot of emphasis. Although each of those presentations that we scored for, there must be a criteria to distinguish one versus other. That's the only reason that we are giving it. But everybody has presented well. I congratulate each one of you. And of course, my best congratulations to those two winners who will be coming to. And only one thing I will request again that I started also in the beginning is to ask questions in a deeper way and the wider participation. That's very important for you among to, that's the indication that you are all interacting. And especially when cross disciplines comes together, the questions that comes from other discipline colleague are going to really improve our thinking. Because oftentimes we can narrow down our thinking in one particular discipline and may not be knowing what's going on in the other discipline. So that's very important, and take care of that part also. And I wish you good luck to all of you. Thank you.
Congratulations to all the presenters, and as told by our co-chairman, Dr. Ravi Krishnan sir, just improve it based on the recommendations given by our chairman, so that you can get a best thesis award, etc. So that you can incorporate in your thesis work and can submit for the best thesis award university, as well as for the JEE best thesis award, etc. Best wishes to all of you.
Thank you so much, panelists, for your wonderful and suggestive ideas. Now, I call upon, now I request Dr. Ravi Krishnan sir, Director, Center for Plant Breeding and Genetics, to honor the guest of honor with a moment. Call upon Mr. Divakar, the first year doctoral scholar, molecular biology and biotechnology, and the student rapporteur, to propose the vote of thanks.
Good evening, everyone. As we come to the end of the oral presentation session on the next generation techniques in crop improvement of 9 AGSC 2024, I take immense pride in expressing my gratitude to everyone. First, I sincerely thank the chairman, Dr. Sindhu Kumar, Professor, Scientist, NIPGR, New Delhi, for enlightening the session with his comprehensive and classic presentation, having a presentation that is catered to all the audience, biotechnology, bioinformatics, and microbiology, genetics, which emphasized on the perspectives and omics approaches and different empirical assays to address the need of our theme, and also finding a database for rising the crop in the vessel of resilience. I extend my gratitude to the co-chairman, Dr. Ravi Krishnan, Director of CPG, for his continuous support and interest in contributing to the welfare of students. I sincerely express my gratitude to Dr. P. Koglade, Madam, Professor and Head of Plant Biotechnology, for accepting our invitation and encouraging us. I thank the faculty rapporteur, Dr. Jayanand sir, Assistant Professor of Bioinformatics, for his active contribution. I thank all the oral presenters for sharing your comfortable research outputs. And I thank everyone for keeping your energy high and putting forth questions and keeping yourselves on about the line. Now, and finally, I thank all the volunteers in making the session a noteworthy one, without whom this wouldn't be possible. Thank you, sir. Thank you, madam.
Thank you so much. Once again, we thank one and all for the grand event. It's absolutely a cherishable one. Now, I request the participants to assemble in the dais for a group photo session.